to play. For example, knowledge of history or study of history it shall be incomplete if there is no carbon-14 dating, if there is no chlorine-37 dating. Who was teaching history, the abundance of iron ore is much more than the abundance of copper. Then why Bronze Age preceded Iron Age?
लिंक छोड़ दीजिए, लिंक चेंज हो जाएगा तब.
There was no reply.
वही, हाँ चलो ठीक है.
You can't explain it without an understanding of chemistry. The answer lies in the Ellingham diagram, the free energy changes, because the temperature which is required to reduce the oxides or the ores of copper is much less than the energy required to reduce the iron ores. And that was the reason. The temperature required for this reduction was much lower, and that could easily be achieved during our earlier era. And that's why it preceded. Once I put a question before a person from history. He was again talking of something about Second World War. My question was, Hitler had controlled almost all or more than 50% of the then Russia. Then why suddenly the troops started retreating? The answers were, okay, because they were too tired, or because it was too cold in the terrain of Russia, so they had to retreat. I told no, there is some chemistry behind it, because Hitler ordered that the German troop should have the dress like the Sipahi of German army should be dressed like an officer of British Army, and that's why putting buttons on the coat and the pants in the dressage of this army, of the Sipahi of the German army was mandatory. And what happened? The chemist knows the property of gamma-tene, and the allotropy, the polymorphism of gamma. Polymorphism of gamma was the reason behind behind the change in the property of the tin button, and that became very soft, and the coats started their coats. They had no option but to unbutton it. Button टूटने लगा, and because of the cold, they had to retreat. So there is chemistry behind any such scope. And one of my Hindi friends, he told, okay, there may be some application of chemistry in history, but where the application of chemistry is in literature. My friend Dr. Bhavesh Pandey we may have some discussion on this topic later on with English. मैंने उनसे कहा, हिंदी वाले से, पता नहीं आप में यहाँ कितना 8 या 9 रस कितना होता है साहित्य में? तो कहेगी हाँ होता है। हमें कि केमिस्ट्री वाले ये बताते हैं कि मस्तिष्क में कौन सी अभिक्रिया हो रही है, किस रसायन का उत्स्रवण हो रहा है, क्या किस तरह के रसायन बन रहे हैं, जिससे वो उस रस की तरफ वो चले जाते हैं और उसी तरह की कृति होती है. There are chemical reactions taking place and everywhere inside the brain and in the neurotransmitters, so many things, signal transduction protein, everything, so many things are there. In similar way, today we have this COVID pandemic, and now researches are going on, and the virologist, because the basic research also, it is incomplete. Take for example any advertisement, in the social perception in any advertisement. अगर आपको घड़ी डिटर्जेंट का दिखाना है कि आधुनिक है, if you have to show a laboratory, the chemistry laboratory, the conical flask or the burette or the beaker or the some colorful solution will be there. And so chemistry is the most versatile, and chemistry is the most standardized subject, because I would give a reference of the IUPAC, the role of IUPAC. There are IUPAP, International Union of Pure and Applied Physics. There are IUPAM, Mathematics, IUPAG, and so on and so forth. You can check the website of ICSU, International Council of Scientific Union. So there are standardizing or monitoring international agencies in many of the subjects, but none of them has succeeded in playing such important role as IUPAC is playing. Even school students are aware of what IUPAC means. Sir, IUPAC nomenclature हम जान रहे हैं, इसमें इथेनॉल बोलते हैं या क्या बोलते हैं सर, ये so IUPAC is known thing, not only because of nomenclature but standardization of any. If you have any dispute, we go to the court of IUPAC. If you have any question on standardization of any measurement or instrument, so for standardization of knowledge, we have divisions of IUPAC. Everybody is aware who is teaching or who is reading chemistry. And there is no other subject which is such standardized, which has undergone such standardization. So we are lucky in getting ourselves associated with chemistry. Chemistry जैसा मानक कोई मुझे नजर नहीं आता है, तो जरा ध्यान दिलाइए कि इसके जैसा मानक विषय अगर राजनीति शास्त्र में है तो कभी प्रश्न पैदा होता है कि अरिस्टो ने ये कहा, प्लेटो ने ये कहा, और अगर सबकी धारणा अलग है तो देयर मे बी conflicts of opinion. In chemistry there cannot be conflict of opinion. Somebody says, okay, हमारी किताब में लिखा है, मेरे पास कॉटन विल्किंसन है, you have been telling that it is Bible of inorganic chemistry, और उसमें क्रोमियम हम देख रहे थे, chromium 2 acetate. So it is a chromium 2 compound stabilized by acetate ion. The sky blue color of sky blue chromium 2 ion has been sterilized, and the scarlet precipitate has come. A scarlet precipitate of chromous acetate has come, and it is a chromium 2 compound stabilized like that. Then you will immediately say, which cotton Wilkinson you are talking about? Is it 66 edition or 1962 edition? Okay, sir, it is second edition, 1966. Then you must know that Geoffrey Wilkinson, who was one of the two authors of that book, got Nobel Prize for intermetallic bonding. Chromium 2 bond is there. That is not a chromium 2 compound of the chromium 2 ion which has been stabilized as such. So the concept, the knowledge, not concept, the knowledge has changed. We are going towards perfection. I remember in school or college days, the Avogadro number that I had remembered was 6.023 और 10 पर 23. See, 6 23 और 10 23 ऐसे करके हम लोग रटे थे. Then somebody saying, okay, I have a book which has been borrowed by my father, from my father, and in that book, that was a standard textbook. पंचानन डे की किताब और किसकी किताब,
हेडफोन लगाइए ना,
वो इसमें लिखा हुआ है कि भाई 23 और 23 है, क्या कि भाई 78 के पहले का होगा. IUPAC क्या अब का परफेक्शन क्या है? 6.022, not 23. So there is perfection and standardization. And now chemistry, besides being most versatile, most standardized, this is one of the fastest growing I'm telling, because the software, the ICT is also advancing very fast. Chemists will have to go for introspection because we are also advancing. But they have, sometimes it appears to me also that the software, the computer science people, they have, their pace is faster than our pace. But it is one of the fastest growing. For example, if you have heat and thermodynamics in physics, the 1932 edition of Heat and Thermodynamics by Saha and Srivastava, it cannot be counted even a single word cannot be said, that cannot be countered. Every word is correct till now. It is almost 88 years now old now, that edition. That I do possess that book because I inherited from my grandfather. He passed on to my father, and I have it with a beautiful black leather cover. So I have preserved it in my shelf. So, but because that is still perfect. But what about the chemistry book? I have 2 volumes of chemistry and elements. My chemistry friends are aware of it. Chemistry and the element by Sedgwick, the wonderful book. But I have preserved it. But that is not up to date. We can't rely on it. That is an outdated thing. So chemistry is the fastest growing. The older edition of book in chemistry is becoming obsolete. It is only for the purpose of history of chemistry. Anyway, so it is one of the fastest growing, one of the versatile, it is one of the most standardized, and one of the most modern, and it is the laboratory. जब भाई ऑर्गेनिक केमिस्ट्री का प्रैक्टिकल हम लोग करते थे 12 घंटे का प्रैक्टिकल, और जयपुर राजस्थान यूनिवर्सिटी और कई जगह तो 18 घंटे का प्रैक्टिकल 3 दिन में होता था। हम लोग कहते थे कि भाई वही रेम्यूनरेशन मिलता है 10 रुपये 8 रुपये कैंडिडेट 3 दिन लेने में 18 घंटे में, जो 3 घंटे में बाकी को मिल जा रहा है। 18 घंटे का 18 घंटे प्रैक्टिकल में और 12 घंटे प्रैक्टिकल में एक पेपर होता था, बिकॉज वी हैड टू स्टैंड इन द लेबोरेटरी, सो द इंस्ट्रूमेंट्स। और जो भी मॉडर्न इसमें दिखाना है, मैं बता रहा था कि किसी एडवर्टाइजमेंट में यू हैव टू शो इन एनी चैनल, इन एनी डिटेक्टिव सीरियल ऑन एनी टीवी, क्या CID और समथिंग लाइक इस तरह का सीरियल जो आता है, उसमें they have to show, if they have to show forensic laboratory or any scientific investigation, they have to show chemistry lab. So it is the experimental science, and it is the experimental science, and इसको किसी चीज में डाल दीजिए वो मॉडर्न हो जाएगा। फिजिक्स में डाल दिया इसको, that becomes chemistry plus physics becomes modern physics. Chemistry plus biology becomes modern biology. If you go to the molecular detail, the structure of the molecules in the biosystem, it becomes modern biology. At many places it is known as modern biology. So we have to adopt modern science by extension. It means modern chemistry to do research for containing and dealing with the coronavirus epidemic, 2019 nCoV, and for the virus has it is seen from 3 angles. The first part is production of the virus. Everybody knows today that it is produced in the infected cell. If the person is infected, the virus cell is being produced in the infected cell within the infected person, and the cell is exploding. The cell, after being killed, is exploding, and it is bursting out viruses, large number of viruses. The production is there. The second step is its migration. So there it needs vector. It can sit on the surface of anything, and then it can move to the target. And so it is how transmission is taking place. So the control, we are thinking of controlling it. We have to think how to stop the production, how to prevent production in the host cell. So how to prevent infection at the primary stage itself. If the person is infected, how to stop or how to reduce the rate of multiplication of the virus, number of viruses. And second, it is being transmitted. How to stop the transmission, or how to kill in between, how to filter, how to kill the virus, make it ineffective. And the third stage, when it reaches a host cell, and the host cell, how to stop it from entry, how to stop the spikes of the virus to get in contact with ACE2 receptor on the surface of the cell, and how to stop this communication. And then after the infection in the biology also, if the cell is getting infected, how to go for a remedial, or how to stop its activity, or how to stop its multiplication, how to prevent the cell, this system from getting killed. So everywhere chemical reactions are there. So from viral, everything from viral structure, the structure, beautiful structure that we are showing every day, it is coming in the newspapers with beautiful spike. It is not as beautiful as it is being put on the newspapers screens on the television. It is not as beautiful. It has some sort of fluxionality in the shape. The RNA, the big molecule inside, it is one of the largest RNA in the virus, we can say. So it is a fluxional molecule, and then the shape is also, it is not so spherical. It is a spherical, nearly 100 nanometer size is there, and so this, if it is being passed, the size should be smaller than 100 nanometers, so that it cannot pass through. So from the structure, viral structure, to pathogenesis, from isolation of vaccine, from isolation of vaccine to therapy. The medicine is also, we are talking about development of antiviral medicine, or how to, or we are trying how to improve the system inside, the immunity, how to improve it everywhere. So basic researchers are depending upon chemistry, from the structure of the virus to the control of the various aspects of virus, the virologists and the clinicians, from understanding and controlling spread of coronavirus, and from all so everywhere the role of chemistry is there. Unless we spread it, we explain it, we can it. Recently, I would just refer to some of the good papers. Only few days back, a good paper. I'm just trying to share the screen. Just let me tell you, it is a good. If I'm able to.
Yes.
Oh, sorry, there is some problem. Anyway, so the PB28 could be a COVID-19 game changer. A good article has come on. It is free access article on in academic in American Chemical Society, ACS Medicinal Chemistry Letters. A good paper. And then another paper in the same in the ACS Chemistry Neuroscience, the SARS-CoV-2, the increasing importance of water filtration against highly pathogenic microbes. Not only this, in future, because we are talking about chemical and biological warfare, how to design and develop method of filtration through which we can get rid of such microbes. So highly pathogenic microbes could be designed. So some good papers, and then what should be how the vaccine should be safer, how to develop and design the vaccine, it should not affect it adversely. So these are many things, and everywhere chemistry is involved. So anyway, with this note, I think I should give you time to, because I had some other engagement also. I wish the webinar a grand success and fruitful discussion, I am sure, because I am finding that Dr. Girish is there, Alam is there, then Ujjwal is also taking interest, and I am finding some of the good participants from different institutions. So we shall surely be make introspection how to develop and how to go for even anything from filtration, from making of masks to wearing PP things, and the protective cover, the things, and from killing the virus during transmission to anywhere. So everywhere chemistry is there, and so naturally for getting for bringing perfection, in any such a step, the role of chemistry is of paramount importance. And with this note, I thank you very much for holding this seminar and conducting it. Thank you, Ujjwal.
Thank you, sir. Yes, I feel a great pleasure to invite. Okay. Uh, yes, sir. This is my distinguished privilege to thank our chief patron who has graced this session with his invaluable words. He has given an insightful speech on this topic. It is hoped that his address will set the tune of the entire proceeding. Thank you. Thank you so much, sir.
Thank you. Thank you. Continue you.
Okay. Now, now I'd like to invite our first distinguished guest speaker, Professor Dr. Rafiqul Islam, sir. Dr. Rafiqul Islam received his PhD degree from Department of Chemistry, University of Kalyani, West Bengal in 2007. After completion of doctoral degree, he pursued his 1 year and 8 months. He pursued his postdoctoral research from University of Witwatersrand, Johannesburg, South Africa, for 1 year and 8 months. After that, Dr. Islam joined as assistant professor at the Department of Chemistry, BIT Misra, Ranchi, and after serving 7 years and 2 months, he moved to Mahatma Gandhi Central University, Motihari, Bihar, and served as associate professor in the Department of Chemistry during the period 14th November 2016 to 13th November 2019. Presently, he is a professor in the Department of Chemistry, MGCU, Motihari, Bihar. His specialization is organic chemistry, and area of research interest is nanomaterials, catalysis, and organic synthesis. Dr. Islam has also membership of societies such as Life Member of Indian Society of Chemists and Biologists India. He supervised to many PhD students. Uh, he has experience of many PhD and MSc students. He has edited two book chapters and published uh, nearly 40 research articles in refereed and peer-reviewed international journals. Also, he uh, completed an ongoing research project of SUBS DST New Delhi. Now, I invite Dr. Rafiqul Islam, sir. Please come and deliver your valuable speech to us, sir. Please.
Thank you, Dr. Ujjal. Good morning to all. Hope am I audible, Dr. Ujjal?
Yes, sir. Proceed. What a minute.
Okay. Good morning to all. Hope all of you are safe and healthy. So at first, I would like to thank the organizing committee of today's webinar, the Exigent challenging role of chemists against COVID-19 pandemic. I am grateful to Professor Ranjit Kumar Verma, sir, Honorable Vice Chancellor of Munger University, Dr. Noel Prasad, Principal, SKR College. Then I would also like to thank Dr. Ujjal for inviting me and giving me an opportunity to present and discuss in today's webinar. So before going to presentation, so I just wanted to let Professor Ranjit Kumar Verma sir that one of your students sir is my senior and good friend, Dr. Professor Kosik Malik. So I had a talk with him, and he told me that whenever you will go, then just inform sir. So okay, sir. So here when I go through the organizing committee, so members from different. It is not only for chemistry. So as a student of chemistry, I try to keep away chemistry in my presentation so that everybody can understand very easily. So I would like to thank the organizing secretaries, Dr. Birendra Pandiji and Dr. Kundan Lal. And our speaker is also Vishwadeep Tripathi is here, my friend, and my friend V. Sandra is also there. So now I'm going to share my presentation. Dr. Ujjwal, is it visible?
No, sir.
Hello?
No, sir. No, not visible.
Dr.
Yes.
Ujjwal,
Yes,
are
you are
you
visible.
visible? Okay, thank you. So we will we discuss about this the challenging roles and responsibilities of chemists. We have to have some overview how the COVID pandemic is. So here we'll show a graph, a figure image. It is a little bit earlier, and I intentionally keep it because we will know that within these few days how the changes is taking place. A few days back, it was in India, the infection is 2.2 million, and now it is 2.83 million. That is, and the date is 44,000 earlier, 1 week before, and it is now 53,978 as of today. And here we will see in the figure, you will see here from the China. From the China it started, the virus started, infection in China, and that is in the late December 2019. And all of you know that the world is now a global village, and that is one of the example that how it spreads to the worldwide. This is the global village. From the Wuhan, from China, it traveled to almost all the country in the world. So it's and all of you know that nowadays, due to this spreading of this COVID-19, that all the universities colleges are closed, laboratories are also closed, and as a faculty members, as a researcher of chemistry, most of the universities, most of the labs are closed, and few labs who are working, few labs in the world are working. So in the next slide, we will discuss whenever we will think about COVID-19, a few things directly came to in our mind. There are some images. You will see the beautiful image of COVID-19. That is the unwelcome guest in our society. That is the COVID-19 images of this. And in our mind, we have that mask, gloves, hand wash, sanitization, all this stuff is there. So as a chemist, so I also think that it is advised to wash our hand frequently. So as a chemist, we should know that actually why we have to wash our hands, and if we wash our hands for 30 seconds, then what will happen basically? What is the chemistry behind it? So here we'll see that why frequent washing of hands recommended. So all of you know this is the coronavirus, the image of coronavirus that is contains some lipid materials and the proteins. Okay, and we have the soap molecules. The chemistry people already know, and for other participants that I am just showing that in soap molecule it is a long chain fatty acid. It's called. One part is attract water, another part is hate water, but loves oil and fats. So when we wash our hands, if some coronavirus virus or other material is present, then what will happen? This soap molecule breaks the genetic material of this coronavirus and forms a micelle here. Micelle means what will happen? All the soap molecules outside and the inside, that is the fragmented part of this COVID-19. So whenever we will wash it with water, then what will happen? This might away from the hand. So this is the way by which we can remove these contaminations in our hand. So this is how, and also we have to know that sanitization, the sanitizer, alcohol is there. Alcohol basically it contains absolute alcohol is 98%. So whenever you will use alcohol, then what will happen? It will evaporate so easily. That's why we have to know that some glycerins, some aloe vera is added. Whenever glycerin or aloe vera is added, then what will happen? It bonds to the alcohol, and then when we will rub it in our hand, it stays at least 20 seconds in our hand, so that within 20 seconds it can destroy the proteins. the coronavirus. So for then we have to know how the virus can spread. So here we will see that is the infected people. If any infected individuals, if coughs or sneeze, then what will happen? The droplets spread. It is travel up to 1 meter and more than also. And in the air also it can go. There is a possibility, so it can go. And then what will happen? Or there are other ways, like the handshake. If we shake hands with the infected people, that this way. Or if we share some materials or bathe some materials, by this way it can infect the people. So in the earlier slide, I have shown you some mask, hand sanitizer. Why we need? Sorry. So here we'll see in droplets. If it is the airborne, if we think it is airborne, then what will happen? In indoor places, the scientists are advised to have a good ventilation so that if there is something, it can remove due to the ventilations. In the droplet transmissions, what we have to do, basically, if this person use a mask and the person, suspectable person, is use a mask, then there will be a very less chances to transmission. In the shake hands is not advisable, and it is also advised to have use a gloves. And also here we will see whenever we will use share some common uses, common things, common materials, then we have to sanitize this first so that we can kill the viruses, and then you can use. So here it is the how can the virus can spread and how we can prevent the spreading. Okay, then we'll have to think about what are the treatments we are preventing by this way, but still the infection is going on, so we need some preventive measures. So what are the preventive measures? So here we have to search some effective treatments. So effective treatments, for an effective treatments, there are three things we need to target. First thing is that keeping the virus is away from entering our cells. This is the first target. Second target is if somehow it enters into the cell, then it prevents replications so that it cannot spread, it cannot grow much more. And then third is we have to target that it can minimize the attack of virus to the other organs. That means there are no effects to the other organs. So these are the three things we have to target whenever we will think about a drug. So next slide we will discuss in the development of drug. So people always used to speak that it is almost 8 months or 9 months. Why the scientists are too late to have a new compounds or new medicines in the market, or new vaccines in the market? But thing is not so simple. We can say in simply that it's a long time and science has developed too much, so we have to have a compound within a very less time. But science is not a magic. We have to have some protocol and we have to follow some way. So here in the drug developments, we can say that as of now, 294 drugs are chosen as lead molecules, and out of these 208 molecules are in clinical human trials. So we'll discuss what is the human trials. So basically, for the development of drug, first we have to prepare the new molecule, new compound in the laboratory, and then it will go from the laboratory to the other to various steps like structure reaction, structure activity relationship, and then animal testing, and then we will go for some clinical trials that we will discuss in the next slide. But here we have to know one thing: as the laboratories, as most of the laboratories are closed, and it takes and to form a new compound, it takes a decade or more time, few years. So what will happen? Scientists all over the world called for wide screening of existing drugs to see if they might work against COVID-19. So that means whatever the drugs we have, that is called repurposed drugs. Whatever the drugs we have, we are using. Scientists are trying to test or investigate their drugs, whether it is effective or not. So here is a timeline how a drug come to the market. So first is the target identification, then target assay construction and screening. Here in the screening, we'll have some biological computational studies that the interactions of the molecule, interactions of the molecule with the proteins, how it binds, and the molecule if you changes some substituents, changes some different molecules,
कैमरा इसमें लेना
how
है,
the interaction changes, and from this
इसमें नेट नहीं है,
we will
नेट
get
इसमें
the
है,
lead molecule. So if
छोड़
we
दीजिए.
use the repurposed medicine, that means the drug already used, then these three steps already gone. That means we no need to have this stuff. Second stuff is there that we, if any medicine which are in use nowadays, if we use them, then what will happen? We know the side effect of that medicine also. That's why it is also easy to use. So then it will go for some clinical trials. That is the phase one, phase two, phase three. I think all of you know whenever you are going through the newspapers nowadays, everything is everywhere you will see. Yes, this vaccine is in phase one, this one is phase three. So basically, phase one is to look for in a small number of molecules, it check for the side effects and how what is the best doses
नमस्ते
for
नहीं है,
use. In this phase two, that is the efficacy, that is the
क्या बोल
efficiency and safety of the molecule
चलाएंगे.
is checked, investigated. And the phase three is done in a several hundred to several thousand most pupils, and it is gives the idea basically that how the drugs is works and in there any side effects in long term. So after that, it get a FDA approval, and then we'll get. That is the steps. So now we know that to have a tool from the very beginning, from the synthesis of the
Like
molecule and to the market, it is almost a decade. So it's not too easy. But now we are using new drug molecule. That's why some
आप नहीं
time
समझ
has
हम
been
क्या कर
less.
रहे हैं, कोई कुछ नहीं
So
कर रहे
in
हैं,
this
बस बोले हैं वीडियो
next
बस.
slide, we'll discuss some molecules which will now
अरे इसको
using
बड़ा
as a
कर देंगे
emergency
भैया,
use
बड़ा हो
for
गया,
during this
कोई
pandemic.
दिक्कत.
These are the drugs molecules. We'll see that is one is umifenovoid, favipiravir, hydroxychloroquine, remdesivir, lopinavir, and ritonavir, methylprednisolone, and dexamethasone. These are the drugs basically approved for emergency use for the severely ill COVID-19 patients. So we'll have some, we'll look after some of these molecules. Then there is the favipiravir. It is an antiviral drugs, and here it is under you will see that is a treatment for influenza. It is already used. Now we know the side effect of this drug, and here is the and in the trade name of Exigent in the Japan, that is the phase three clinical trial is going on for COVID-19 patient. In India also it is approved for emergency use, and Glenmark Pharmaceuticals that is making this heavy favipiravir in the name of Feviflu for the treatment of mild to moderate COVID-19 patient in India. So this is the Feviflu, and we will get in the market. So that is for, but this is for emergency use, not for a normal use. And another drug is umfenovoid. It is also an antiviral drug that is marketed in Russia under the brand name Arbidol, and it is also used in China for the treatment of influenza. So people are now thinking that whether this medicine is effective against COVID-19 or not. That's why the treatment and clinical trials are also going on about this molecule. Here, the hydroxychloroquine sulfate. Whenever we will talk about hydroxychloroquine sulfate, then we remember that what happened in India. Basically, India first banned the export of this hydroxychloroquine sulfate. Then what happens that President of United States, Donald Trump, called our honorable Prime Minister Narendra Modi ji for delivering this hydroxychloroquine sulfate to the US so that they can use as an emergency use. So this is also used as an emergency for the hospitalist patient. Then remdesivir is also an antiviral drugs. That is also the clinical studies trials are going on in the East China and United States. So I think all of you know the Gilead Pharmaceuticals. So they are checking whether it is effective for COVID-19 or not. So this is the remdesivir molecule. And then we'll have prednisolone. This is also a medicine that is called glucocorticoid. The chemistry people will know. It is also investigated whether it is safe or whether it is effective in the treatment of novel coronavirus in China. So the next medicine is the dexamethasone. All of you know, I think it is basically used for some asthmatic asthma people. That means when they have some problem in the respiratory systems, then this medicine is used as SOS. This is a cheap and very widely used steroid that reduce the death risk among the COVID-19 patient up to a third. So that's why in the University of England they have investigating this one, but it is not useful for less serious illness. Only for severely ill people they are using. Here it. Then we'll have this one. One is lopinavir and ritonavir. It is also a combination of drugs. Basically, it used for the HIV virus, named Caletra. So this drug that is the mixed with a combination with Tamiflu and in Thailand. And what happens once severely they have shown that when this cocktail used in Bangkok, then what happened? One patient is recovered from the severely suffering COVID-19. So that means clinical investigation of this drug is also going on worldwide. Then this is one compound that is called Sheikhpura, that is commonly known as EIDD-281. It is also an antiviral drug and is orally active. Basically, it is used for the treatment of influenza. So now what will happen, as we will see here, that it is a hydroxycitidine molecule and derivatives, and it has an antiviral actions. How it happens that it introduces of copying error during the RNA replications. So by this way also the COVID maybe that the replications may not take place. So that's why the activity against the COVID-19 is also going on about this molecule. So these are the molecules. Basically, these are the drugs using in the clinical trials to find a efficient or effective medicines in the therapeutic drugs in the market. Then what happens? It is the vaccines. Development of vaccines. So there is a good news also. The good news is that everybody knows, I think, the 12th August 2020, the first time in the world, a vaccine is registered in Russia for COVID-19. But the problem is that they have skipped the clinical trials, one more clinical. Within 2 months, they have registered this one. So before discussing this one, first we'll go how many, what are the vaccines? A vaccine basically trains the immune system, trusted source, to recognize and attack the virus when it encounters it. That means when we have a pathogen into our body, then the vaccine will induce the immune system, and then they kill the virus, attack the virus. So here we will see there is the these are the vaccines. These are the vaccines that is in clinical trials. And the fourth runner is Oxford one, Oxford University vaccine, then CanSino, then Moderna. These are the three is on the track, and we have to know something about the vaccine trial process. Drug trial process and vaccine trial process, there are something different, but for phase 1, phase 2, phase 3 is there. So here we will see preclinical study that is done in animals. So whenever we have the vaccines, then what will happen? It tested on animals whether they have produced a immune response and what are the effect, is there any side effects to the animal. After that, if it think it is safe, then it is goes to the phase one with several people, several healthy people. Then they will say, they will check whether it is safe and how the immunity is responsive and what are the effective doses. Then in the phase one, in the phase two clinical trial, it is several hundred people, and here we'll know what are the safety best doses amount, how many shots we have to have for the vaccine to kill the virus, and how the immune response is. And phase three is the final phase where several thousands people are volunteered and injected with the vaccine, and we and investigated how does it works and whether there are some side effects or not. So this is how the vaccine trial process is gone. And after that, there will be an approval and it will come to the market. So here will be it is in the Russia Gamaleya Institute and Russian Defense Ministry. Defense Ministry have introduced this new COVID-19 named as Sputnik 5. But the problem is that they have registered this virus within 2 months of testing. They have skipped a phase three trial. They have not yet done the phase three. After phase one and phase two, they have introduced this one. So that's why scientific communities around the world have is suspecting and whether it will be effective or not, whether there will be some side effects or not, because they have not also shared the data with the WHO. So here we'll talk about the vaccines, some vaccines. So here in the world, around the globe, all total 150. I think from today we can say it is around 160 vaccines candidates are there, and out of these 29 are in human trials. Human trial means they are in phase one, phase two, or phase three. So more than 28 candidate vaccines are being tested in clinical trials for in people. So first one we'll discuss, that is the Moderna one. Moderna, that is the US company. They are using messenger RNA, and they have already performed the clinical trial 1 and 2, and they shows that they have produced antibodies and they are safe. Now FDA approved that they can go for the phase three clinical trial. So after completing the phase two clinical trial in July, they have begun the phase three clinical trials of their vaccine. So we are hopeful if phase three, if it is fine, then it finally comes to the market after the regulatory approval. Then we'll have the vaccines, that is the University of Oxford in England. That is the front runner vaccine in the world. So there is a possibility, high possibility, and
क्या कुछ व्यवस्था किया है
we
कैंसर
may
का
positive on it.
कर रहा है
So here already they have already reported
क्या कर रहा है
the
वो
data
ले ले
of clinical trials
हाँ
phase one and two, and they have
स्टार्ट
showed
अभी नहीं
that
किया है ना
they have also the safety, immunogenicity and efficacy of this
अच्छा
vaccine,
मिस्त्री
and
की
they are
आया है क्या
well safe.
हाँ
So now the phase two
अच्छा
and phase three clinical
दिक्कत होगा
trials
तो ऊपर में
have
चलेंगे
started in
इसको आप
UK,
लीजिएगा
US,
पावर
Brazil,
लेंगे
and South
ऐसे ही लेट
Africa.
पहुंच जाएंगे हम अपना कैमरा बंद
So
कर
I
लेंगे
was in South Africa in that
नहीं नहीं मेरा
university, and in that
मेरा वीडियो
medical
बंद है कोई
college,
दिक्कत
in which medical
नहीं
college
ले ले चल
the
जाएंगे
clinical two, three
आप इस
trials
चाबी को
of this
रखिए ये
Oxford
चाबी मेरा
virus
वाला
is
है ना
going
यहाँ
on.
पर मैंने
And
चाबी
in
दिया
India,
इसको
Serum
बंद कर
Institute
दीजिएगा
of India have tied
ये
up
गेट
with AstraZeneca
ये तो मेरा है
and the University of Oxford
नहीं उनका चाबी कहाँ
to
है
study the clinical trial phase 2
नहीं
and phase
इसी
3
में
in
इसी में
India. The Indian government approved competent authority
ये वाला
already
चाबी है
approved
ठीक है
for their clinical trials, and hopefully by this month they will start. They have already processed and
अच्छा
they
कोई बात
will
नहीं चलो
start the phase 2, phase 3 clinical trial. And if it is successful, then under the brand name of Covishield, it is SII, Serum Institute of India, they will launched in the market if
हाँ
it
उसका
is
काम नहीं है, छोड़ दीजिए।
positive,
खाली
if
तैयार
get
रहिएगा
positive
जानने के लिए।
results. Then another is Cancino Biologics in China. They have also developed a vaccine,
ये देखिए तो क्या हो
and
गया?
they have their phase 2 trial is already over, and phase 3 trial, phase 3 clinical trial
हाँ
that is will be going on Saudi Arabia and Pakistan. But
वैक्सीन
as like Russia, China also granted the patent of this first COVID-19 vaccine in China to Cancino. We have already given the patent, but still the phase 3 trial have to
जो अभी
perform.
रिकॉर्ड हुआ।
So it's basically a dangerous thing because without the phase 3, if someone use, there may be some risk. So here, one other is the Novavax. That is, it is an Australian company. They have also started this phase 1 and phase 2 clinical trials of these vaccines. And all of we know that whenever we see Operation Rap Speed, that is related to US, they are US and they are funding some companies to produce vaccines. And these companies, Novavax, it is also funded by the Operation Rap Speed of US for their clinical trial of phase 3. And if they are successful,
आप कहीं
they
नहीं
will
जाइए,
deliver
कहीं
100
नहीं
million
जाइए।
doses as early as late as to the 2020 or in 2021. Then another two vaccines that we have to know, that is the CoronaVac, that is the Sinovac. This is also a Chinese company. But here what will happen, they have already got the results of phase 1 and phase 2, and they are so positive results. That's why phase 3 clinical trial they have started in Brazil, Indonesia, and Bangladesh. They have already started in Brazil and Indonesia, and Bangladesh they will be preparing. Then another vaccine is by Sinopharm. It is a China-based pharmaceutical vaccines, and here they have already completed the phase 1 and phase 2 clinical trials, and they are successful. Now, the phase 3 clinical trial they are performing in the UAE in Abu Dhabi. But there is one interesting point is there. Under the hashtag that is for humanity, 15,000 participants, 15,000 participants in the phase 3 trial have been registered and already started the clinical trials in UAE. And the beautiful thing is that among these 15,000 participants, 107 nationalities, that is not people from 107 countries have participated. So there is a possibility is there. If this vaccine is successful in clinical trial 3, then what will happen? As these most countries, if persons are participating,
पूरा कोरोना के
then
बारे में दे रहा है
that
ना।
country can also use this one. Otherwise, there are some
नहीं, हम
techniques
उसकी
in
बात
there.
नहीं कर रहे, हम
If
मैटर की बात
any
बोल रहे हैं,
vaccine is
मैटर
developed in
मैटर जो
somewhere
उसकी बात।
and you want to use in some other places, then there's maybe a phase 2 or phase 3 clinical trial in that country. So then we'll go for our indigenous COVID-19 vaccine. That is under the brand name of Covaxin. So it is developed
छोड़
by
दीजिए,
Bharat Biotech
नेट इसमें चलिए ना है तो फिर
and in the collaboration with ICMR, the National Institute of Virology. So it is a indigenous, and here what will happen that the phase 1 and phase 3 clinical trial have already started, and the initial data shows that the vaccine is safe. Now after
वो तो 80-90 तक गया था,
getting the
अभी काम
complete
रहा है आ
phase
जाएगा।
1 phase 2
अब तो काम
data
रहता है ना,
results, then
बीसी
they
पानी
will
सब हट
proceed
गया,
for
कॉलेज वाला
phase
सब
3
हट गया, साइड ले
clinical
लिया।
trial. And another vaccine is that Zydus Cadila also is working in one vaccine. In the 15th August Independence Day celebrations of our Honorable Prime Minister Narendra Modi ji, we were told that 3 vaccines, 1, 2, more than 2, that is 3 vaccines are working in India. One is the Covaxin, that is developed by Bharat Biotech. Another that is in phase 1 and phase 2. Zydus Cadila vaccine, that is also in phase 1 and phase 2. And then another is the Serum Institute of Technology India, which in collaboration with AstraZeneca and the University of Oxford, that is also in phase 2 and phase 3. Final trial is going on in India. These are the 3 vaccines candidate which are most probably maybe success and will get. Then After all, finally, what happened that we need a vaccines very desperately, and we have to sanitize the global the coronavirus, and we need the
यहाँ पे
effective
extension से
coronavirus.
line नहीं आ सकता इधर
We have this,
इसी रूम में
I can say that
ए
we have vaccines, but the real issue is that how safe it is, the efficacy, the efficacy. 10 more vaccines are
इस रूम
in
में कहीं पलक वलक नहीं
phase
है
2 and phase 3 trials. We may hope we have a vaccine for COVID-19 very soon, by December 2020 or early 2021. But the issue is the mass vaccinations of a country like India. So we have 1.3 billion
देखिए नहीं कहीं पलक
populations.
बैकअप वाला
So
लैपटॉप लेके यहीं चल जाएंगे ना इसलिए
we can just skip this one. Okay.
ओके
The presentation is not
Okay.
now, right? Presentation is going on. Okay.
Okay. Sir, presentation is finished? Okay. Thank you,
So,
sir.
1.3 billion people, so we need a huge vaccinations. So it may take time. Even we have a vaccine by December. At least I think it will take another 1 year to complete the mass vaccination process in India. Although Russia and China have registered the vaccines, but their efficiency, efficacy, and side effects, there is a question mark for this. That's why till then we have to wait for the vaccine. Uh, until then, what happened? We have to follow the guidelines of WHO and the Ministry of Health and Family Affairs from time to
ले जाइए
time.
सर
That is the wash our hands, cover
इसमें
a
ले
cough
जाइए
or
ना
sneeze. That is use of mask. Don't touch anywhere in bare heart.
ऊपर में स्टेशन
Keep
ना है
social distancing.
अच्छा ले
Stay at home means whenever you require you go out. Otherwise, stay indoor and stay healthy. And if
इसका
you
भी
have
लंबा
the
है ना आगे
problem,
तक चल
then
जाएगा
you will call a doctor and see a doctor. So
हम्म।
there is a hope that by this
देखिए ना बाहर से कितना
get
जा रहा है
Sir, hello.
यस
and
एनी क्वेरी
Hello.
यस
Rafiq sir. Rafiq sir.
यस एनी क्वेश्चन क्वेरीज
Yeah, I have a question that Rafiq sir can please explain me what is the cellular organization of coronavirus?
cellular organization.
Of coronavirus, sir.
That is a structure.
Sir, cellular organization of coronavirus. Please explain me.
In which slide you have seen?
It's like a nutrient.
Sorry.
It's like a nutrient or a sir protein.
In this,
Your voice is not clear, sir.
जल्दी कीजिए
okay, now it's all clear. Now it's clear.
Yeah. This is clear.
So you will yes.
Okay, sir.
I also have a question, sir.
Okay.
Rafiq, sir, I also have a question.
Okay, please.
Sir, what is the chemistry behind the co-vaccines which the ICMRA has been produced today, 2 days ago? Co-vaccines. What is the chemistry behind?
That is the Covaxin, right?
Yeah.
Actually, I am not a vaccine expert. I am from chemistry. So anyway, I will tell you that Bharat Biotech and ICMR, okay, and they
Yes,
have
sir.
collaborated. They have produced this vaccine.
यस
And basically, I am not. Didn't went in deep of what is vaccine and how it is generated and how it prepared, because to make these lectures for everyone, just I have given the overview basically. So if we go inside, then that only chemistry people will understand, that other people will not understand.
Okay, sir.
Okay. And this vaccine is in phase 1, phase 2 trial. Okay.
Okay, okay, sir.
Okay, any queries else?
Yes, good afternoon, sir.
Yes, absolutely.
Hello. Yes, sir. Good afternoon, sir. Like, do you have any idea how long this COVID virus stay outside the body? Because some people say 2.5, some say 2 days. So, any idea?
It's somewhat controversial basically. Whenever you research articles, you will see in some places sometimes that means if it is a wooden place, it takes 7 hours. In a steel, it can stay in 2 days. Like this way, it is somewhat controversial. But anyway, if
Hello.
in sunlight, then it cannot stay more than 2, I think 4 to 5 hours.
Yes.
Now,
Okay. Thank you.
okay. If any question later we can discuss. We have another speaker. Thank you, sir. Thank you so much for your kind, for your wonderful lecture. I think all participants are have learned many things from your slide and presentation. You have discussed many vaccines which is useful for COVID-19, and definitely this is highly informative. So thank you again, sir, for your nice presentation. Thank you so much, sir.
Okay, thank you, Dr. Rajal. Thank you.
Thank you so much, sir. Now we have another. Distinguished guest speakers. Now, Dr. Vishwadeepak Tripathi, sir. Now I would like to invite our second distinguished guest speaker, Dr. Vishwadeepak Tripathi, sir. Dr. Vishwadeepak Tripathi has received his PhD degree in chemistry from CDRI Lucknow in 2012. After completion of doctoral research, he served as senior research associate during 2012 to 2015 at Research and Development API Zydus Catla Healthcare Limited, Vadodara, Gujarat, India. After that, he moved to LCC CNRS, Toulouse, France, and he worked there as Sheikhpura postdoctoral fellow during 2015 to 2017. After that, in 2017, Dr. Tripathi joined as assistant professor and head of the Department of Chemistry at MK College, Lahriyasarai in Lalit Narayan Mishra University, Darbhanga, Bihar. He has published 29 research articles in high-impact international journals and 5 patents. Dr. Tripathi has been awarded by many awards and honors such as Best Presentation Award by Mahatma Gandhi Central University, Motihari in year 2020, Young Scientist Award by Lucknow Christian College in year 2018, CDRI Incentive Award by CDRI Lucknow in 2011. He is a member of many professional bodies such as life member of the Association of Carbohydrate Chemists and Technologists of India, life member of Association of Chemistry Teachers, Mumbai. Now I invite Dr. Vishwadeepak Tripathi, sir. Please come and deliver your valuable speech to us, sir, please.
Thank you very much, sir. Am I audible to all of you?
Yes.
Thank you very much, sir, for your nice introduction. My greetings to all of you. And I do not want to miss this opportunity to show my gratitude for Honorable Vice Chancellor, sir, I am highly inspired by him because he is also in the field of chemistry. So, and respected principal sir, respected faculty members, dear friends, and dear students. I am Dr. V.D. Tripathi, a very young researcher and teacher, and let me congratulate to Dr. Ujjwal to organize this wonderful topic in the webinar for role of chemists, because as in the very inaugural session, Honorable Vice Chancellor explained the role of chemistry. I cannot stop myself to sharing one of my experience. I was involved in examination, and I was having the duty of checking the documents. So one of the students come to me and ask, where is this department? And I asked him, where you are going to take the examination? He told me, I am going to take in mathematics honors. So I asked him, why in mathematics honors? He talked me for 10 minutes. Then after he go and he took admission in chemistry honors. So this is the power of chemistry. Anyway, so my previous experience is almost 12 to 13 years related to chemistry and drug discovery. So I think why not to share my experience with my young colleagues and friends, and to let them know the process of drug discovery, and the people who are asking every day and every hour, we are going to get any medicine for COVID, because this is the biggest question. So once people should understand that finding a new medicine is not an easy task. You are going to find a pin in haystack, and you don't know how much time it will take. So my experience was very robust, from very basic research to API research, then in manufacturing, then again in basic research, and now I am involved in teaching. So sharing of my experience will be very interesting to listen for my young colleagues. So now I will share my screen.
लो इंफ्रास्ट्रक्चर में इस टाइप से किया बहुत बढ़िया कमेंट कम दिया, बहुत अच्छा कमेंट दिया है सर, नाइस वन, वेलकम तो देखने को मिल रहा है, इस पर क्या करते हैं?
So is my presentation is visible, sir?
एक presentation यहाँ से
Ujjwal sir,
yes.
Sorry, my presentation is visible or?
Yes, it is visible.
Yes, sir.
Thank you.
Yes, sir.
So,
yes sir, yes.
First reason why we are doing this webinar is this tiny devil. Why I am telling this tiny devil? Because it is very small, as the sir told, the size of around 100 nanometer. But why it is devil? Because it has stopped all the world. All the boundaries are closed. Labs are closed. Locks in the offices. And we are doing all the things on laptop. So it has changed the life in each and every way. Not only the one aspect of life. Our way of living, way of eating, way of reading, education, economy, every sector. So this is nothing less than a devil. So now the question arises, how can we get rid of this devil, and how much our chemistry is contributing? So let me tell you, in next few slides, you will be convinced that this is the chemistry which is working in each and every step to fighting this devil. Now the severity of this COVID-19 infection, it can be imagined or understand in the way that within the very short span of time, it has closed all the world. What is the special with this virus? Or does this virus was existing earlier, or this is something very new? So when people in expertise in virology, they go in the depth and they find out that these coronaviruses are not very new. They have also caused a pandemic and epidemic earlier in the history, but there is something very new with this COVID virus. That is named as SARS-CoV-2, SARS. SARS-CoV-2. SARS was the disease that appears around 2002, and it covers almost more than 50 countries in the world. But what special thing with this COVID-19 virus is the presence of, as you can see here, the spike glycoprotein. And this spike glycoprotein make it very, very dangerous. Like when it touch any surfaces, and the surfaces of some living system like human being, our skin. So these glycoproteins has a very special affinity towards the surface receptors. Like you can see here in this slide, there is a surface receptor, and it has a very fast binding affinity with this as proteins. And when you see whatever drugs we have in our hand, that drugs act at the surface of the virus. And surface of the virus, it enters and destroys the genetic material. But what is special with this virus? It has this spike protein. So once the drug molecule is going to the virus, it has some difficulty to enter in the virus or to touch the surface of virus. It has some barrier around the sphere of the virus. So this is a very special feature, and this is the reason why most of the drugs which are in antiviral are not very sure medicine for this COVID-19. So now in the first slide, you understand, see this affinity of this S protein with the receptor is also an application of chemistry, because without chemistry, it is how the virus is binding to the receptor. So this comes with the application of chemistry. And now once virus is attached with the surface of body or living system, now what is the fate of virus? So whenever in foreign particle or pathogen enters in the body, it has see here, it is attaching with the receptor of the body, then the fusion takes place. And when the virus gets inside the body, it goes under translation to increase the genetic material, and it creates more and more viruses. So this is the cycle when it will go in the body, and after the genetic material replicate, and it will increase the viruses. So this cycle will go on to make dangerous viruses, to make the key points virus on increase the in the body, and this is the reason why they will. So, once we have entered the problem is like pandemic. Pandemic means it covered all the globe. So what comes in our mind or researcher mind, how to get rid of this problem? Once the problem related with the health hazard, we have the two options, whether prevention or cure. Wherever you go, whether you go in the prevention, whether you go in the cure, everywhere there is application of chemistry. So role of chemist is very, very essential. Why? You can see in some coming slides that prevention, what is the main difference between prevention and cure? Prevention is a primary. Anyone can do by his own prevention. So prevention is an easy way. It is not very costly. No one is going to ask to give the money to him. You can use your money according to you. It lives the healthy life, means it one side it is curing, it is prevention the viral infection, and other side it is making you more healthy for all of your life. It has a happy family. But when it comes to the cure means this killing the pathogen, remove the pathogen. This process is not easy. This is complex. This is very expensive. In few slides, you will see how much expensive this is. Even you cannot imagine. And this is very time consuming. This is very stressful. So this prevention is always better than cure. but both of them have the application of chemistry. How? You see, what are the preventing measures for coronavirus? We say that wash your hand, and Professor Rafiq have explained very nicely in his lecture that why washing is very important, because washing is you are washing with a soap which is going to form the micelle, and thus micelle is disrupting the viral genetic material, and so it is very difficult to, and it is also disrupting the attachment of this S protein with the skin or receptor. So viruses go out from your skin, it is not going outside. We have to cover ourselves, like what was there is a PPE kit, personal protective equipment. So that is also made up of all the polymers. All thing is chemistry, and we cooked thoroughly everything to remove the pathogen. We have to keep the social distancing. So these are the preventing measures for this. What effect these preventing measures do in our body? The first thing is preventing measures prepare our body for a new war. New war means that it is not the last virus on the earth, the COVID. There may be some other virus can come in future, or this virus can also mutate to have new weapons. So by prevention, we are preparing ourselves, we are preparing our body, and we are boosting our immune system. And these herbal remedies are very easy and powerful weapons in the nature which we can use against these evils. Now the question comes, is the cure? So as I told you in earlier slide, that cure is not something which is very fast and easy. Cure is something, here you see this medicine is going to kill the virus or inactivate the virus or inactivate the infection. So to understand how this cure comes, you will have to understand that what is the process of drug discovery, and how what are the different stages that a molecule has to go for to come in the market. People say that phase 1 clinical trial, phase 2 clinical trial, phase 3 clinical trial. What are these clinical trials, and what is happening with the molecule in this clinical trial? How much money it costs, how much time it takes. Once you will understand all these things, then you will have an idea that how the molecule comes in the market from the lab, and how this cure is available to fight against diseases. So if medicinal chemistry, again, see, this is the root behind the cure, behind the discovering of new medicine, vaccine, whatever, this medicinal chemistry. What is this medicinal chemistry? Medicinal chemistry is not only chemistry, it has mixing of various branches, interdisciplinary, that is organic chemistry, biochemistry, computational chemistry, pharmacognosy, pharmacology, statistics, physical chemistry, everything. So once you can easily understand that one person cannot be the master of each field. So it is very interrelated topic, interdisciplinary. We should have a very strong team with experts from all the field to finalize the molecule which we can use as a weapon against COVID-19. So how the medicinal chemistry evolves? See, this is nowadays we have to devote a lot of time to discover a drug, but it was not the case every time. In 1950s, when the science was evolving, we were not having so many branches, and the synthetic chemistry was there. We synthesize a molecule, and then we test the activity. It is done. Our molecule is active, launch in market. But slowly, regulations are coming in the picture. Various regulatory bodies are formed, various different branches are formed, which makes the process complex. In 1960s, it was the evolution of biochemistry. Then after that, the rise of physical organic chemistry make the process more complex. then molecular biology, then 1980s after the discovery of computer and software, we get the computational chemistry, that is quantitative structure activity relationship, QSAR. It makes the process again more time taking. And in 90s there was a flood of different branches: computational chemistry, combinatorial chemistry, chemistry of automation, data, chemical informatics, everything. So all these things making the process more time taking, more costly and more complex. This is the reason why launching the molecule in the market is not an easy task for a pharma company. After 2000, the era of genomics has come, and nowadays we know all the genome of viruses and we can design the vaccine. So what lies ahead? How it is going? So you see the power of the pathogen. Power of pathogen. Don't imagine that pathogen is very small. See how fast it is multiplying in 1 hour, in 2 hours, in 4 hours, in 6 hours. And in 24 hours, even you cannot count how much cells are formed for a pathogen bacteria in the body. And what is the speed of production of medicine? So both of things cannot be compared. So if we think that we are very powerful, no, more powerful organisms lies on the earth. And this proved by this COVID-19 infection, that when all the world was busy with doing the nuclear power test and everything, and all the boundaries are closed by this single virus. This is the roadmap. This is my favorite slide to explain you that you see this is the long way. You start from here and you are moving a long way to go here in the market. How much obstacles are there in between? Because the journey starts from the idea. See, this bulb is rising. So idea is rising in the in our mind, that, and now there is no question of idea because idea is given by the virus. We have to fight against virus. So what is the next? Next is the project team meeting, and we have a team with the chemists, with the biologists, with the physicists, and with the toxicologist. Everyone will sit on the like a round table conference. They will discuss, and now the question arise for target identification. Supposing like coronaviruses, we have different targeted like proteases, because in viral infection, when you will see the proteases have a very crucial role. So we fix the target. Then we will design the molecule by computational chemistry, that what kind of molecule will be very suitable for this target. And once we will design the molecule, now we will have in our mind, not in our hand, that this kind of molecule can be active, can be very good to kill the coronavirus. So now the start of chemistry comes in the picture to synthesize the molecule. And a very big team of chemists or PhD students will involve in the laboratory for a synthesis of a library. Library means thousands of compounds. And nowadays the machines are available, robotic arms are available, which can do the thousands of experiments on a single day. And even as a chemist, most of you know that how much time taking is the process of synthesis, multi-step synthesis, and to characterize your molecule. So suppose once you finish all these things, the synthesis and characterization of molecule, the next step is to test these molecule against the bacteria or virus culture. That is in vitro analysis. And in vitro analysis, we are very lucky and everything goes in our favor. We get some molecules which are active in vitro testing. So right now we are in very beginning, but you can imagine how much work is there. Once we have a molecule, we will go to test the toxicity, that whether our molecule is toxic or not. Toxicity will be normal cell. So we have the molecule which is showing in vitro activity and which is not toxic. Then we will go to do in vivo experiments. In vivo experiments in lower animal models like rat and mouse. So suppose we are still lucky that our molecule is still showing very good results in vivo testing. We will go to study the ADME parameter,
सोनी सब सोना बोल रहा है
absorption, excretion,
अच्छा वो लिंक तुमको भेजे उसको follow
metabolism,
कर
and distribution, ADME parameter. And once we get to know that, okay, this molecule is excreting from the body, this is not doing any hazardous metabolite in the body, then we can go further with this. So if still we are lucky, everything is going in our favor, then we will go for animal model, testing on animals. They can be rabbit, they can be gerbils, they are lower animal model. Then we will go in higher animal model like monkeys. And if all these things are going okay, very, very costly processes. One animal model even can cost you more than 2 lakhs. So if everything is again going on our favor, we get everything right, then our molecule will finish the preclinical stage. Preclinical still. And all the results will be filed in the thesis or dossier, and that will be presented at DCGI, Drug Controller General of India. And this the office of DCGI will review the dossier and give us the permission to perform the clinical trial. Now, 1/3 of the journey is finished. Still 2/3 is remaining. If we get the permission of clinical trial, because clinical trial is not an easy task. It has 4 stages. 4. Phase 1, phase 2, phase 3, and post-marketing surveillance. What is phase 1? Phase 1 is very risky, very difficult. 95% molecule fails in phase 1 clinical trial. Suppose if everything is going in our favor and we get the permission for clinical trial, then the phase 1 clinical trial will start. Phase 1 clinical trial is the clinical trial where molecule is tested against healthy volunteer. Healthy, not disease. Suppose we have some vaccine or molecule for COVID-19, then that molecule will be tested on a healthy person, not on the person who is suffering from COVID. So this is not an easy task to educate the volunteers for phase 1 clinical trial. It is difficult, but we are lucky. India is a very premier country in the field of drug discovery and drug development, manufacturing, everything. We have the facility of clinical trials at different centers. In the phase 1 clinical trial, there is a team of doctors which patient is under observation in the team of doctors. So once phase 1 clinical trial finished, we have a good results, then we get the permission for phase 2 clinical trial. 2 days back, I was seeing in the news that one of the vaccine from Bharat Pharma has get the permission for phase 2 clinical trial. So this is the stage of phase 2 clinical trial. Now in phase 2, the molecule will be tested for diseased person, not healthy volunteer. And if phase 2 is covered, then it will go in phase 3. And in phase 3, it will be tested on large scale, various cities, hospitals in various cities. And if still everything is going well, we are going to file NDA, new drug application. That okay, we are going to file new drug and we will get the patent for that molecule. Now we are safe. No one is going to touch our molecule. Then we get the FDA approval, Food and Drug Administration, and our molecule will launch in the market. But still, post-marketing surveillance is remaining. After the post-marketing surveillance clearance, that molecule will be available. So you can see how long this journey, how complicated this journey, and all this process normally take at least 10 years for development and billions of dollars. At any stage, if you fail, you lose all the money. So this is the reason why Indian pharma companies hesitate to work on the new molecule. Because India is famous for a generic market. So, but now slowly we are growing in the field of drug discovery. So drug discovery is process I told you in last slide. Target identification, target validation, in silico modeling, synthesis, screening, lead generation. Then it comes under the development process like preclinical and clinical, and then human trial. So as you go further from target identification to phase 3, you see the dollars are increasing. And here, how much dollar are, suppose we get failed at any stage of clinical trial, a lot of money is wasted. So drug discovery is a very long, very risky and highly interdisciplinary process. Okay, so now the question comes that no one knowing that we are going to encounter with COVID-19. So if coronaviruses came in our life, what can we do? So again, it is the application of chemistry, that what can we do? Okay, we cannot find a new weapon. We will use whatever we have in our hand. So whatever drug we are already have, we will test that molecule. This phenomenon is known as repurposing. One molecule, that darunavir or remdesivir, that was made for AIDS virus, but it is showing effect in coronavirus also. So this is the repurposing of drug. At one side, people are making the new drug and vaccine. At one side, doctors are using repurposing of drug, which is curing the life. So how the repurposing of drug works, you see here at different targets like chloroquine and nafamostat, lamtinib, it acts at the endocytosis stage. This chloroquine at the binding of the virus with the receptor, and this lopinavir, ritonavir are working at the inhibition of proteases, this 3CL and PL proteases. So at different stage, different molecules are working, like this remdesivir and favipiravir. This works at the replication of genetic material. So it disrupts the replication, and here you can see these antibodies, that tocilizumab and sarilizumab. So these are the different, this was a very nice article, it was published in Science. You can find it on internet to see the detailed mechanism of action of all the drug at a single place where I found so. With the idea of this repurposing, we get some weapons in our arsenal, like and these drugs are classified on the basis of their targets, how these drugs act, and at which stage the drug are so going to act. Like the molecules which are viral dependent RNA polymerases, these molecules inhibit the RNA polymerases. These are this galidesivir, darunavir, and you see this complex structure, stereo center, all these things, and you can imagine how typical synthesis of this molecule is. So now you can understand the role of chemistry, because chemistry, without chemistry, even one cannot think the synthesis of these molecules. These are synthetic molecules, not natural. So, and the chloroquine, as you know, this chloroquine was like a sanjeevani in this time of COVID-19 pandemic, when it was, we feel very proud that this molecule was firstly synthesized by Indian person, that Praful Chandra, and these molecule also work as inhibition of polymerases, RNA polymerases. This boloxavir, favipiravir, encyclovir, pimodivir, these drugs already exist and they were using in the clinics. We are again using against the COVID-19. This ribavirin, this remdesivir, this is very famous molecule and gets approval also to use against the COVID-19 infection. Then these baricitinib and chloroquine inhibit the viral infections. This darunavir and lopinavir against the viral proteases. Ritonavir and nitazoxanide, it also inhibit the COVID virus by inhibiting the viral protein expressions. The virus, this umefenovir, also used for HIV virus, and it also helpful in disturbing the attachment of virus on the surface of the body. So this is the power of chemistry. See the synthesis of darunavir, how complicated is that. We have this primary amine. This primary amine is going to open this oxidant ring. Again, after opening the oxidant ring, this is the substitution of this sulfonyl chloride and this reduction of this nitro group to amine, and then this reaction with trifluoroacetic acid to deprotect the bulk deprotection. So all these are very complex or tedious chemical steps for synthesis of darunavir, and this can be only possible by application of chemistry. So now the story is moving forward. We suppose we had the molecule, we get some drug or a molecule which is killing the virus or inhibiting the disease. After getting the molecule, what will be the next step? So sometimes in debates in TV, we see that suppose we get the vaccine for virus, then how the 130 crore people will vaccine will be available for all the people. So again, the problem is not going to end. So once we have the molecule, then the role comes in the picture of pharmaceutical industries, the manufacturing plants. We have the process in a laboratory, we did all the things, but now we have to manufacture the molecule. And this time we are not manufacturing some polymer.
आवाज नहीं आ रहा है, आवाज नेट तो है।
What happened, sir? Hello? Hello?
Yes,
Audio is not working, Ujjwal, sir.
yes. I think some technical problem. We are looking.
Oh,
Technical problem. Sound is not coming. Hello. हाँ. Yes. Hello. Yes, I think there is a technical problem. So we are checking after some time. Please wait. Uh,
There is some technical problem. Please be with us. We will resume this webinar soon.
शायद डिस्कनेक्ट हो गया।
I think Deepak sir is disconnected, no?
Yes, I'm calling. I'm calling maybe
Yes,
power
Ujjwal
on.
sir, he is
Maybe cell
disconnected
phone power off.
probably.
Maybe.
I'm also
Or
seeing that he is not in the sport. So you may kindly invite our third speaker if there is some major problem. Ujjwal sir, are you listening me?
You're calling is not answering.
Ujjwal sir,
yes, yes, yes. I'm calling to.
Ujjwal sir,
Yes, yes, yes,
are you listening me, sir?
yes, yes.
He is
I'm
not
listening.
on. He is not on the board, sir.
Yes, I think he's disconnected.
Yeah, sir. So
So
please invite our third speaker, sir, please, so that session is not interrupted.
1 minute. Okay. Yes, I think Dr. Tripathi sir has disconnected. So he has presented a very nice talk. So I'd like to give thank him. He has presented many medicinal compounds and which is highly important for uh, highly important drugs like galidicivir, darunavir, chloroquine. Many compounds are people are knowing. So, very wonderful talk he has presented. Now, I would like to invite our last speaker, Dr.
I'm sorry, sir.
Okay, you are connected.
Doctor sir. Actually, my data was finished and I was not knowing when the meeting
Okay,
was
continue,
there.
sir. Continue. I thought you completed.
Which
Continue.
slide was going on when it was disconnected, sir?
Okay. Just proceed, sir. Proceed.
Which slide was going on when it was disconnected?
Middle one.
Okay.
Tripathi sir, it was a roadmap of research roadmap.
Oh, roadmap. This was a slide, sir.
No, sir. This is not that slide. That was Danur Veer, sir.
After this one, yes.
Next slide, sir. Next slide.
Next.
Sir, it was about Darovir.
Yes.
Okay. This was a slide.
Yeah.
Okay, so
Yes,
with this
sir.
slide, I was
Continue.
I was telling that this is the very complex synthesis of darunavir, and this can be achieved only with the help of chemistry. So once we have the molecule in our hand, and we can use this molecule for that COVID infection, then how this molecule can be available for all of us, for 130 crore of people in India. So this is a big task. And now the role comes in picture of pharmaceutical industries. How the pharmaceutical industries are involved in producing the molecule, because once the pharmaceutical industry are manufacturing the molecule at bulk level, then it will be only available for the large scale, at the large scale for persons. So in the pharmaceutical industry, now the picture comes of analytical development of or analysis. So once molecule is synthesized, then we have to analyze the purity of molecule, how much impurity, how much other things are present in the molecule to affect the activity of that molecule. For that, we have different instruments in the laboratory like this HPLC, as you can see here, high pressure liquid chromatography, and this works on the principle of chemistry. The chemistry is essential. For this moisture analyzer, we calculate the percentage moisture present in the molecule, and that works on the chemistry, chemical arithmetic. This gas chromatographic principle works for on the chemistry principle. These NMR spectrometer are professional chemists have frequently encountered these instrument. This XRD instrument is very popular in industry to characterize the molecule and match with the reported one. This NMR spectrometer is used for analyze the different kind of nucleus. So if I tell the role of chemistry, time is unlimited to tell the role, because role is very vast. But with the drug development, chemistry is important at each and every step, like target identification, designing of molecule, synthesis of drug, characterization of molecule, manufacturing, quality check, impurity analysis. All these steps are revolving around the chemistry. So chemistry plays a central role. So chemistry has a key role in the play of understanding everything from viral structure to pathogenesis, isolation of vaccine and therapies as well, development of materials and techniques caused by basic researchers, virologists and clinicians. And the fundamental research is to understand the molecule that govern the viral structure, and the function can accelerate the discovery of means to treat the prevent infection. So finally it comes to end. Thank you all for your patience. I am sorry for the inconvenience, and I will be very happy if you have any question.
Yes, if any question you can ask. Any question?
Especially from our young students.
Sir. Hello.
Yes, yes.
Yes, your voice is coming.
Sir, can you please explain me about the in vitro analysis?
In vitro analysis is analysis where you are doing the analysis outside the organism body. In vitro analysis, you are culturing the artificial culture of pathogen. It can be bacteria, it can be nematode, but that is the culture. That culture is artificial, not natural. You are doing testing outside the body. That is in vitro. It consumes low cost, less money. So first we clear the obstacle of in vitro, then we go for in vivo, because in vivo testing is very costly.
Okay, sir. I can just understand.
Yes. Any question else? Okay, thank you. Thank you, sir. You have presented a very nice lecture, and I hope all the participants have learned many things from your presentation. You have discussed many drugs like chloroquine, hydrochloroquine. I think in India everybody is aware about this compound.
Mr. Ujwal,
Yes.
Mr. Ujwal,
Yes, yes.
I am Dr. Sanjeev Kumar Jha.
Yes, tell
I
me.
have a question to Dr. Tripathi sir.
Yes.
Tripathi sir, am I audible?
Please, sir, please ask. Please.
Sir, it is said that it is a RNA virus and mutating, sir, very fast. So how could, sir, it be possible to claim for the any vaccine if it is mutating very fast?
See, vaccine is something which is very different from the drug molecule, because vaccine is formed by the calculation of the genome of the virus. What kind of genome this virus is, and now when we know the genome of virus, this is RNA virus, so we can make this biological, these known as biologics. We can make the vaccine which counter the genome of virus. So whatever type of virus you have, you can be sure that you will have the vaccine within certain span of time, and maximum it will take the 1 year or around 1 and 5 years. Because you can have vaccine, but you cannot sure that once you will have the vaccine, the virus will be like the original. Because virus has the power to mutate. It is mutating very fast, and it can also change the genome. Then we have to change the vaccine. So this is the complementary processes are going.
Okay, sir. Okay, thank you, sir. Thank you, sir.
Thank you very much.
Any more question?
Excuse me, sir. Excuse me. Yes, I want to ask also. Good afternoon, sir. Sir, I'm Aditi Krishna from MRM College, Barbigha.
Okay.
Sir, my question is here that what is the role of darunavir in COVID-19? COVID-19 that drug. As you were telling that darunavir synthesis is quite long and large and it's very complicated.
Yes. Actually, I have a very close relation with darunavir because when I was in industry, I was doing a project for the developing scale-up synthesis of darunavir, and I know that how difficult it is because sometimes we face the days when we have nothing in our hand and working for 1 week. So darunavir is very unstable API. This molecule is not very stable. But your question was regarding that what it is doing for COVID-19. So the answer lies in one slide. You can see that darunavir is. I was showing here the target of darunavir is. One second. It was something. Yes, the target of darunavir is inhibit the viral RNA-dependent polymerases. See, polymerases are very crucial enzymes for viral life cycles. They are responsible for the some phosphate attachment and phosphate release. So these proteases are very important for virus life cycle to grow. So once you will have something which inhibit the proteases, virus is already going to inhibit. So role of darunavir is to inhibit the viral RNA-dependent proteases. Now to understand the RNA-dependent proteases, you have to read a little bit of targets of COVID-19. Yeah, I will explain you personally. It is very interesting.
Thank you, sir. Sir, one more question. Sir,
Yes, sure.
as our webinar topic is that what is the role of the chemist in COVID-19? So, sir, many efforts are being done in all over the world in developing the COVID-19 anti-drug. So, till now, there is no result found.
See, you are asking this question because you are asking this question because you are not a professional medicinal chemist. Because this is what I explained here. To discover a drug, it is not something which you revolve the magic stick and you have the molecule. It is a very rational process, very rational. And sure, we will have in our hand the vaccine, and it will come, but it will take time. It will take all the barriers and all these things.
Yes. Thank you,
Yes,
sir.
good afternoon.
Okay, sir. Thank you.
One last question. Sir, my name is Neelam, research scholar from BN Mandal University, Madhya. Sir, I want to know, can this vaccine be administered to a patient as well as a healthy, and how does it work inside the body? I mean, how does it function?
Okay. See, the vaccine is something which you will be, which you will give to patient as well as healthy. Both you can give, because vaccine is synthesized for healthy being also. Once the vaccine will be available in India, that vaccine will be given to all the people in India, because this will generate the immunity in your body, and when after vaccine you will encounter with the coronavirus, nothing is going to happen with your body.
Yes, sir. So, I mean, what it does inside the body? Does it also mutate or react with this COVID?
Yes, sure, because COVID has a special power to mutate. because you see, like SARS, severe, that SARS in 2002 and MERS in South Africa, that all has coronavirus, but they are mutating very fast. So it is very much possible that it can mutate. And might be you have heard that at some places, like in USA, we have a more deaths with COVID-19, and at some places we have less deaths with COVID-19.
Yes.
So
Yes.
there are the different strains of viruses. There are.
Yes, okay.
So,
Dear participants, we have last third speaker, so we can discuss later. So time is very limit. So thank you, sir. Thank you so much. You have presented a very amazing lecture and discussed about different type of medicines we learned, and I hope all participants got many wonderful ideas about the drugs related to COVID and other diseases. Thank you so much. Thank you so much, sir.
Thank you, sir. Thank you.
Now, I would like to invite our third and last guest speaker, Dr. Grish Chandra sir. Dr. Grish Chandra sir completed his BSc and MSc from Vinoba Bhave University, Hazaribagh, and then he obtained his PhD from IIT Bombay under the supervision of Professor Vishwakarma Singh. In 2008, he immediately joined Ranchi University, Ranchi, as an assistant professor. He then moved to Eva Women's University, South Korea, to pursue postdoctoral work with Professor Lak Sin Jeong. There, he worked on the synthesis of modified nucleosides for different therapeutic cancers and viral diseases like HCV, hepatitis C virus, HIV, and anti-cancer drugs. Presently, he is a senior assistant professor at the Department of Chemistry, Center, University of South Bihar, Gaya. His major research interests are in the areas of organic synthesis of biologically important molecules, development of methodology for fluorination reactions, and development of the chemistry of different types of cyclohexadienone. He completed one major project, and recently 3 projects have been approved from reputed agencies. He has published 17 reputed research articles in international journals such as Journals of Medicinal Chemistry, Organic Chemistry, etc. Now, I invite Dr. Grish Chandra sir, please come and deliver your speech to us. Sir, please proceed.
Thank you, sir, and greeting to all of you. I am feeling very happy and very privileged to share some of my informations and knowledge about chemistry, which is basically useful to check out COVID-19. So first, I would like to thank Dr. Ujjwal Kumar for organizing this kind of a wonderful webinar, where basically different kinds of students having different background are basically taking participation and enhancing their knowledge about particularly medicinal chemistry. Medicinal chemistry, particularly in Bihar area, is very unknown kind of a field. So if you get some knowledge about medicinal chemistry, then it will be always wonderful. Medicinal chemistry has a lot of applications and lot of scope, particularly for getting different kinds of jobs. Okay, so before I start, let us just share my PPT. So just wait. So can you see my PPT?
Yes, sir. Visible.
Yeah, okay. Thank you. So as basically Professor or Honorable Vice Chancellor Dr. Verma sir mentioned that basically Ujjwal Kumar basically chosen very good topic, and this is basically related to basically updation of knowledge in chemistry to COVID-19 pandemic. Okay, so before I start, first I would like to thank once again Dr. Ujjwal Kumar Bhagat for inviting me for sharing my knowledge and information related to COVID-19. and organizing this kind of a webinar, particularly in Bihar area. Okay, and I also thank another organizing secretary and committee member for setting this kind of a webinar. I once again basically thank Dr. Naval Prasad to providing this kind of a platform for organizing this webinar. And finally, I would like to thank our honorable, basically Vice Chancellor Professor Ranjit Kumar Verma for giving opportunity to young mind to basically deliver his knowledge or deliver his background knowledge to just highlight some chemistry background to COVID-19. Okay. So before I just go once again, I would like to basically acknowledge different resources which basically I took different kinds of photographs, graphs and structures from internet and different books and different material. So here I basically acknowledge all of them. Okay. So during last 6 months, I will say 6 months, we are basically facing a very huge crisis for human being. We are basically getting problem due to a virus that is called coronavirus disease 19, or in short it is called COVID-19, and due to which the whole world is now become static. We are unable to do our regular work. We are unable to do a study. We are basically facing a lot of problems. Okay. So question is, what is coronavirus? So many of already know coronavirus is basically a large family of virus which may cause illness in animals or humans. In humans, several coronaviruses are known to cause respiratory infections ranging from common cold to more severe disease. So if you try to recognize, what is the basically appearance of coronavirus, then one important thing is that you will feel cold and you will have severe fever and you will have a throat infections. So if you have this kind of a thing, then you take seriously and try to consult a doctor. Okay. So respiratory syndrome, which is called MERS, and another one is basically severe acute respiratory syndrome. These two are basically developed due to coronavirus, and due to that, in last decade, many people got died. So these are basically well-known viruses, but one new virus we basically witnessed in last 1 year, and that is called COVID-19, or in another word, this is called Corona COVID-19. Okay. So this is very unique in the sense that the first time this kind of virus is recognized by a human being, and we are basically never infected or never got any kind of a problem due to this Corona 19 or COVID-19 disease. Okay. So if you go through the data available, then up to basically 8 lakh people got died throughout the whole world, and up to 2.2 million people got infected. So this is not a small number. This is a very big number, and scientists are basically thinking that this number will be very, very huge in next 6 months. So they are thinking that up to 25 lakh people throughout the world will die due to this COVID-19. Okay. So coronavirus or the COVID-19 particularly is very novel, but the pandemic caused by different virus are not new. So if you go through the history of human society, then you find that there are many pandemic caused by virus, and millions of people basically died due to infections of different kinds of virus. So here I feel to summarize few of them, particularly from the last 500 years, which basically created a problem for human society, and sometimes it was so severe that it was thought that it will kill the whole human society. So the first significance one is basically during the century 1346 to 1353 that causes 20 million people basically death in Europe only. 20 million people, and this is due to the basically plague. So this was basically historically very famous, and I will say one important significance of this plague epidemic. Okay, after 2 century in 1665 to 1666, once again 1 million people got died in London city itself, and that was 1/4 of the populations of London city. To see the devastating nature of this plague. Okay, there are many data are available, but the most important and most rememberable for human society is basically the Spanish flu, which basically started in 1918, and it causes death of 10 crore of people, and it was estimated that 1/3 populations of world was infected. 1/3 population. It was thought that if human population would not show the unity or the eagerness to fail this attack of virus, then maybe human being got extinct from this earth. So continuously there are many more pandemic caused by human society, and we are basically a younger generation. We did not see this devastating basically pandemic, but in last 10 years we observed or we witnessed 3 important pandemics. Although the toll of death is lower as compared to the previous one, but the significance one is basically H1N1 swine flu, which basically caused 2 million people to die. This is once again due to basically virus. Another one was basically West African Ebola, that is causes 11,000 people to die. And now presently we have a totally new kind of a virus, and it is basically killing more than or basically it is continuously killing many people. And today I just listened one news in India itself. We have the highest number of COVID cases, that is 70,000, that is in yesterday. So that is a huge number. The problem about the COVID-19 is that at present we don't have a significant or impressive vaccine or medicine to combat this COVID-19. मैं सुनाई दे रहा हूँ ना?
Yes, sir. Yes, audible, sir.
Yes, sir.
Yes, sir.
Yes, sir. Yes, sir. Proceed, sir.
So let us continue. I specifically want to mention once again the Spanish flu. Okay, so this basically started after just World War I end. Okay, World War end. So it was thought that the people died due to Spanish flu. It was basically more than World War I. So you can just think about the severity of this virus. Okay, so total 10 crore people died during this Spanish flu. So my whole presentation related to this webinar I just want to mention what kind of information or what kind of significant scientific contribution we made after this kind of pandemic. Okay, so specifically I took this example, and here I want to mention few of the significant contributions from different scientific community, which basically not make made basically understanding properly different virus, but also we developed different kinds of vaccines and medicines. Okay, so basically I just here want to mention that every natural challenge, just like a virus infection, basically give us a opportunity to understand more properly or more in the molecular level. Okay, so day by day is basically we are improving our understanding of scientific knowledge, and due to that, if you compare the death toll due to different virus infections, then you feel that we are basically improving our hospital facilities, drug facilities, vaccine facilities, and our health are also improving. We are getting different kinds of knowledge, awareness, and due to that, overall death is basically lesser as compared to the previous one. So here I just try to summarize few words, which basically signifies the contributions of different scientific community after this Spanish flu. Virus basically was discovered long back in 1882, but after spread of Spanish flu, scientists basically more properly try to understand the basically virus, and the most significant result came that they were
आया गया
basically
आया
able
गया
to
लेकिन ठीक है अरे
synthesize
बहुत अच्छा
virus
परफॉर्मेंस रहा
in lab.
हाँ
They
शिवम
developed a tool to synthesize
लेक्चर सुन
virus
रहा तू मेरा
in lab. They basically developed
अच्छा
a
अच्छा ठीक है
method by
आज तो सुन रहा है घर
using
में
chicken egg, and
ठीक
this
है तुम्हारे
method is
पापा
still
हाँ
we
हाँ बोलो
are using to basically synthesize virus in lab.
अभी चल ही
So
रहा
this
है
is the one
तब
important contribution after
नेट खत्म हो गया चलो
Spanish flu
ठीक है
pandemic. And not only we
ठीक
basically developed a
मेरा
basically
भाई ने
method for
रजिस्टर किया है
making the
फर्स्ट
virus,
पे है
but
रजिस्ट्रेशन करा
also
एंटर
we
एंटर करा
developed a
ग्रेजुएशन में है
different kinds
तो
of
सर्टिफिकेट देना था
method
इससे नंबर
to
करना
make
पहले
the
रजिस्ट्रेशन करो
vaccine.
फिर चैनल
Once
आओ
again, I will
तो मिल
mention
गया
here that
गवर्नमेंट में क्या
the
कहा कि लिंक कहाँ
method
से
we are
लाए लिंक
basically
में पड़ जाए
using at present
तरीका
to
अगर
synthesize
पाकिस्तान है तो ठीक है
to make
मेरा
the
भाई भी
vaccine
ज्वाइन किया
are
रजिस्ट्रेशन मेरा भाई
basically
है
already developed
रजिस्टर उसको भी ज्वाइन किया
during
फैजान भी है
1930
ओके
after the pandemic of
ऐसे
COVID-19.
इसमें जो आएगा जो रजिस्ट्रेशन किया
And
वही
during
आएगा
this
आएगा
time also,
नील कमल
one
भगत
important
मेरा अपना जुर्म भाई है
significant
सगा भाई उसको
discovery
रजिस्ट्रेशन
came,
किया कल
that is
उधर लिस्ट में आया अब उसको सर्टिफिकेट तब
in
देना वो सीरियल नंबर
1884
ही
it
सब
was
नहीं है
discovered
कोई
that
बात नहीं कभी चेक हुआ तो हम टाइप कर देंगे
the responsible for genetic code. So this was basically the one important
ये भी आसाम से
contribution.
बोल रहे हैं एक
Okay.
लखनऊ से बोले एक अभी
So
यहाँ
there are
से
many. So at the present it is not
ठीक
good
है लोग
to
अभी
just
ज्वाइन कर
use
रहा है
all
मैंने
kinds
फिल्म
of
मारिया
information in this
तमिलनाडु
platform.
तमिलनाडु का कोई
But here I
पार्टिसिपेंट
just want to highlight
है कितना
few.
बुरा हो
Okay. So now
अटेंडेंस
question
जा रहा है कभी
here,
100 गया
what is
कभी
the
80 गया 90 गया अभी
contributions of
53 को ऑनलाइन है 59
so
चेक
here
कर रहा है
I will
बहुत
mention
फिल्म
that
बहुत
during
10
this
5 20 से
time
तो बहुत ठीक है ना
people started
यूट्यूब का अलग
relying
से नहीं है
on
यूट्यूब
the
भी
synthetic
लगभग 10 मिनट
medicine.
वहाँ भी होगा अपने
Basically,
कोई
after
रजिस्ट्रेशन किया सब बोका सब
development of penicillin
घूम
was
रहा है
basically
इनके जलन हो
synthesized
रहा
by
है जेलस
chance in 1929.
यदि
Okay,
भगवान को
and it was
समझ कर
found that this was
जैसा
basically a
कंटिन्यू हो रहा है
wonderful
ना
drug
इस
for
जगह पर आने वाले समय में
against bacteria.
हम अपने
Okay,
संभवतः
so but the
छोड़ देंगे
process of
जहाँ तक
making the
कृष्ण बाबू का
penicillin was
सोच था
not very effective.
लड़ाई
So
मतलब
synthetic
जेसी
chemists
सर के पास मेरा
basically
केस बना कितना भी जाके
basically jumped
हम लड़ते हैं
and
नाम
tried
समझ
to
रही है
make this
अब
compound in
पहचान
a
में आ
huge
गया ना
amount.
देखिए सब 10 बार
So
नाम नहीं लेना
penicillin is basically one of the
जितना
good examples.
टीचर सभी दूर
Another
से
good example is basically quinine. So these two drugs,
कैमरा देख लेते हैं कैमरा
although developed in
देखिए
basically
सबको थैंक यू
during
बोलने के लिए बात
1930
ले ले
to
करके ई कर रहे हैं ना
1950
क्या थैंक यू
and
बोले
was considered one of the
उसे
basically
इंट्रोडक्शन ठीक
precious
नहीं है
drugs in last
नहीं नहीं
century.
ठीक
You can just think that these two drugs basically saved thousands of people during World War II. So basically, once again, I want to mention that always nature
आ गया
gives
हमारे
us
साथ
different kinds of challenge, but human being,
30 मिनट
particularly
कितना
scientists,
समोसा लग सकता
are
है 23
always
समोसा
eager
लगा करके
and alert to just try to understand
25
those
समोसा
kinds of challenge
एक
and
एक
try to
समोसा
give
25 हुआ
basically
200 रुपये
uh,
बिल
the
एक एक
basically the
अरेंज करवा सकते हैं सामने में
molecules or
तो मेरा
particularly or
समोसा
the
आपके अंदर में है जो 25 रुपये
scientific
करवा दीजिए
knowledge to basically hamper those
कितना
kinds of
पैसा देंगे
challenges. Okay, so here the importance
देखिए
of
हमारे पास जो
chemistry
है
and
आप
particularly synthetic
लगाइएगा फिर आपको
chemistry
सामने देखिए
for drug development is very
अरेंज
important.
कीजिए
as mentioned by Tripathi sir, drug development process is not a very easy task. Now, most of people are thinking about coronavirus and they are waiting eagerly when basically drugs will come. But I will say here, at the process of drug making is, since is not very easy, so you have to at least wait for at least 3-4 years to have a suitable drugs to basically counter COVID-19. Okay, so the question is, what is the role of synthetic organic chemistry? Obviously, synthetic organic chemists make or devise a process to synthesize these kinds of compound to make in abundant amount and to deliver these compounds to different kinds of patients and alleviate different kinds of disease. So question is, what is the process for making these kinds of drugs? So basically there are three methods available by which we can make these kinds of drug in abundant amount. The first one is total synthesis. What is total synthesis? Total synthesis is basically you just start from a very cheap starting material and construct different functional group or different carbon-carbon bond and make our target molecules. Okay, so this process is also lengthy and very costly. Another method is semi-synthesis. Semi-synthesis is basically partial isolations from nature and partial basically synthesis in lab. And third is also very important, that is a large scale fermentation process. Okay, so we do just fermentations of different kinds of natural entity and try to extract our main ingredient and use those ingredients as a drugs. Okay, so here I just comment that synthetic chemists both in academic and industrial laboratory played very important role in addressing serious human health crisis on a global scale. What kind of disease we will have, the synthetic chemist will have a very important role. Ultimately, the synthetic chemist will have to synthesize the compound in the bulk scale and deliver to market for alleviating different kinds of disease. Okay. So now question is one question arises, one general question generally people ask, if nature have these kinds of important molecules is able to alleviate different kinds of disease, then what is the need to synthesize these kinds of molecules? To answer these questions, I will just take one very important example, and this is very important molecule that is called taxol. So this compound is very important, and this compound is used for the treatment of cancer. So this is a chemotherapeutic agent for the treatment of different kinds of basically cancer. Okay, so this compound was basically isolated from the bark of Pacific yew tree. So here, if you are able to see, then you can see the tree. This is a Pacific yew tree. So it was considered that if you isolate bark and extract this molecule, then that molecule will be useful for the treatment of cancer. But if you see the data by cutting 100 years old tree. Okay, so consider if any cancer patient need 200 milligram for per day, then you have to cut at least 3 trees to get the regular dose from nature. Obviously, this is not a good way to basically get the medicines from the nature. And what is the solution? Solution is to just solution is to just synthesize this kind of a complex molecule in lab. Okay, so if you see the complexity of this molecule, this is not a very straightforward to just go in lab and just make this molecule. This is very, very complex molecule. If you see, there are many numbers of chiral center And if you change any one of the chiral center, then property of this molecule will get changed. So if you want a exact therapeutic use of this molecule, you have to exactly synthesize this compound in lab. So synthesis is not a very easy task, as basically Tripathi sir mentioned in his presentation. So, and if you synthesize this kind of a compound, then obviously the cost of this compound will be very, very high. So I remember in last 10 years, during 2010, the cost of one medicine of Taxol was basically 12,000 per tablet. 12,000 per tablet. Very costly. The question arises, this is the real cost of this medicine? No. Sometimes what happened, basically company put extra cost for their development. They put extra cost for future development of drugs. They put extra cost to recover the loss during the development of previous
सर नहीं
drugs.
है
Okay, so due to that, obviously the cost will be very, very high. Okay, so fortunately, this is the medicines for cancer, but still we are waiting for drugs for COVID-19. Okay, so now question is what remedies are available for COVID-19? Our vaccines are available? Obviously, we have answered no. Our medicines are available? And once again, we have answered no.
इनके बाद
So
आप
curing method
नेक्स्ट बोल करके तो
we don't
प्रोग्राम
have
के
at
बोर्ड ऑफ के लिए
present.
आप तैयार रहिएगा
Then what is the solution? Then solution you
हाँ
have
आप
to
लाइने पर रहिएगा सर
focus on the prevention.
हाँ ठीक है बस
Prevention of spreading of coronavirus. So what is the main criteria for prevention? The
सर
prevention,
इनके जैसे लेक्चर खत्म होगा
you
इनको
have to
थैंक यू बोल
do
देंगे
the
हम
social distancing,
उसके
you have
बाद
to maintain the
उसके बाद
social
क्या बोलेंगे
distancing, and you have to maintain the self quarantine. Quarantine is not a new thing in this scenario. Long back, people basically cut the spread of different virus by just self quarantine. Just remember, during 1660, when plague was basically spread in Europe, there was no any kind of medicine. The question arises how they basically solved that problem. The problem was solved by doing self quarantine. One famous example related to quarantine is due to Newton. Newton was basically quarantined during 1660, and he utilized those time to develop a different physics theory, and one important theory that is called falling apple observation that led to the concept of gravitational theory of Newton. Okay, so WHO also basically mentioned different type of different precautions measure to take to basically cut the spread of coronavirus. And here I just want to mention few of them. The one important one is basically just clean your hands and wash properly with good soap, or more important by different or good quality of sanitizer. Maintain at least 1 meter or 3 feet distance. Avoid going to crowded places. Avoid touching eyes, nose, and mouth. And most important one is stay always in home and try to do self isolation. So the question is, we cannot do long term prevention. We cannot basically isolate ourselves in home. Okay, so once we have to develop some kind of drugs or basically vaccine to basically combat this coronavirus. So already Tripathi sir and Rafiq sir already mentioned the different stages of development of drug development. I will not go in detail, but I will just mention few of them. Basically, drug development you can divide into three categories. The first one is called discovery stage. Another is basically development stage, and third stage is called review stage. So consider if I am an academician, then most probably I will do the discovery stage. Why? I will mention in short time. Okay. So in discovery stage, what we generally do, we just either randomly or by using the bioinformatics knowledge, we try to synthesize different kinds of molecules, different kinds of organic compounds. And if you have a sufficient number or sufficient quantity of those compounds, then you just collaborate with biologists. They basically screen those compounds against drug targets. Drug targets could be basically enzymes, receptor, carbohydrates, uh, nucleoside, and many, many more. Okay, so these processes either you can do in vitro or in vivo. So after getting some good result through this screening process, you can go through some animal screening, and still if you found some good compound, then you can go through human clinical trial. But as I just mentioned that if I am an academician, then the process of doing human clinical trial is not suitable for me because this is a very costly process. So generally, academicians try to get patent at this stage, that is a discovery stage, and if compounds are very worthy, then you can just sell to different company. Then they will do further studies of development stage, that is a human clinical trial stage. So Tripatis are already mentioned different steps and different loopholes of this human clinical trial stage. The first stage is very, very important, and in the first stage, you need a very healthy people. So getting healthy people is not a very easy task. If someone tell me that I made some compound, can you check this compound in your body whether this is safe or not? Then most of people will deny. Okay, so we have to convince, we have to give a lot of money to those people to lure, so that they will basically get agree to do one clinical trial. Okay, so after this one, if you are successful in one clinical trial, then you have to do phase 2 and phase 3. In phase 2, basically, in phase 2, you have to basically take the real patients. The number of patients is very limited, and you have to study their basically efficacy of that drugs. And if you successful, then you have to go to the phase 3 stage. So what is the difference between phase 2 and phase 3? Phase 2 is basically limited to few hundreds of basically patients. But in phase 3, we have to spend the screening to in many people, different kinds of people, different kinds of ethnical people. If possible, you have to screen the drug from different countries. Okay, so if you are successful, then you approach to FDA, they will give permissions to launch in market. But once again, at least 1 year, you have to keep that drug in surveillance. And if there is some problem, then FDA will withdraw that drug and everything will go on. So if you see, the whole process is basically average we need 13 to 15 years. If you are lucky, then only 5 to 6 years will be sufficient. But in average, 13 to 15 year process is basically to bring in any new compound to the market. And if you see the cost of developing drug, that is a very huge amount, 2.6 billion. This is the average quantity. So Tripathi sir already mentioned that due to this amount, many Indian people are not eager to take a chance to develop a molecule for a drug. They just focus on a generic drug and focus making generic drug. Okay, so I will just skip because many things have been already discussed about human trials. Now, question is. We are ready for development of drug from the zero stage? Then obviously answer will be no, because we need medicine or vaccine very urgently. We can't wait for next 10 years or 15 years to get the medicine for COVID-19. Then question is, what is the solution? Then there are three kinds of solution. Particularly, I will say two kinds of solution. We just use repurposing of drugs. Okay, and second one is basically a study already synthesized library of compounds for getting medicines against COVID-19. And third one is not related to present scenario, but this is also a very important role, that is altering the present viral drugs for improvement. So one by one, I'll just discuss already concept of repurposing is basically discussed. So I will just go in fast. Okay. So repurposing is basically is a strategy for identifying new uses of approach for investigational drugs that are outside the scope of the original medicinal indication. In short, we can just say that we are already using some drugs, okay, and we try to basically investigate whether a particular drug is able to basically solve the problem of COVID-19 or any other disease or not. Okay, then question arises: what is the benefits of doing this one? So there are various advantages or benefits. First one is the safety issue. Since drugs are already in use, so we don't have to check the phase 1 clinical trial, because we already know that this drug is suitable for use for human being. Okay. Second is basically a risk of failure, because we have already known compound, we can make those compounds in abundant quantity. So risk of failure is very, very low. Okay. We also save a lot of money for repurposing purpose. Okay. And we also save a lot of time. Okay. So by using this approach, basically many drugs basically utilized for using for different kinds of disease. So here I took two important examples. The first one is aspirin. This is a very famous molecule, and most of people basically use this medicine for alleviating the pain. When this compound was launched by Bayer in 1899, it was used for analgesic or just alleviate pain. Okay, and during 1980, it was found that basically this compound is very, very useful to coagulate blood. So this is basically antiplatelet aggregation. So this compound, which is basically very, very cheap medicine, we can also use for coagulating blood. Okay, and most use of this by different doctors, that if you have a heart attack, just take aspirin and chew. So the severity of heart attack will low down. Okay, so this is the good example of repurposing of drugs. Another important example is thalidomide. Thalidomide is very famous, and basically I will say having a very bad name in the medicinal chemistry, or basically in drug society. Why? When basically it was launched in Germany, it was basically used for treatment of basically morning sickness in the pregnant woman. But it was observed within 1 year that after taking this medicine, women deliver baby having a deformed in nature. Some baby had only one eye, some baby had only one leg, some baby had only one hand. So within 1 year, German government withdraw this medicine, but after a few years, it was found that this medicine is very, very useful to treat leprosy. So this is also a good example for repurposing of drug. Okay, so for COVID-19, so basically scientists have been investigating various drugs that may be repurposed to fight COVID-19. If you go to the literature, more than 200 drugs basically scientists have been investigating Okay, so for any medicines or particularly for COVID-19, the focus area is to just stop the replications of virus. Okay, Tripathi sir already mentioned different stages of replications and survival of virus inside the human being. Okay, so if you have a understanding of replications and survival inside the human being, that gives you idea about designing new drugs. Okay, although here we are not designing new drugs, we are just trying to repurpose already available drugs for COVID-19. Okay, and second thing is that if possible, try to aim to control the symptoms of the disease. Okay, so here I just took 15 most important, which are basically showing a significant result, and I'm not going to discuss in detail because both speaker already mentioned the use of these drugs in present scenario. And the most significant I just want to mention is basically ivermectin. So ivermectin is basically used for anti-parasitic drugs. So this is a very common drug, and I hope that most of people use this drug during their childhood. When you have infections of parasitic, then basically doctor recommend this drug to just relieve the pain caused by different parasites. Okay, so this compound was basically showing a very promising result, and this drugs basically was studying by one Australian scientist, and I hope that in future this drug will come in market and show some promising results. Okay, so these are few most which basically already mentioned by both speakers. Here, these are basically different kinds of proteins which are basically used as an antibody to treat different kinds of diseases. So this avastin is basically used to treat cancer. Uh, this one is basically, these two are basically used for the treatment of arthritis. So this is a bulk molecule, big molecules. Okay. These are basically different kinds of proteins. Okay, so these are also are under the process of study to just investigate against COVID-19. Okay, so now question here: what is the responsibility of chemists to check out coronavirus? So already mentioned by Tripathi sir and Rafiq sir, chemist has a lot of scope and a lot of work to just do the study to synthesize the molecule and many. So here I just mentioned few of them. Chemist can basically try to understand the structures of virus. If you know the structures of virus, then designing new kinds of drugs or designing new kinds of vaccines will become very easy. So during the drug development for any kinds of disease, the first stage is basically know the drug target. You should know about the different kinds of virus or bacteria or parasite. If you know the structural property, then the half process becomes very easy. Another contribution from chemist is developing different kinds of testing technologies. If you remember, when basically this COVID-19 spread, many people came and basically developed different kinds of technology to basically test COVID-19 in a fractions of, or I will say within 1 hour or 2 hours. Okay, so this is a very important contribution from biologists and chemists. Another important contribution could be development of vaccines, improvement of chemistry which require a smaller sample size, research and development of medicines, development of sanitizer and protecting equipment for doctor and society. Okay, so in many ways, basically chemistry can contribute to check against
पूरा नाम लिया सक्सेस हो गया मेरा काम.
Now, more specifically, I want to just go through the contribution of synthetic organisms.
नहीं हाँ उस
Okay.
प्रेजेंटेशन दे रहे हैं
So I already mentioned that without synthetic organic
गिरीश
chemistry,
चंद्रा सर
you cannot realize any drugs to the market. Okay, so you should have a specialist synthetic organic chemist. They can make smaller quantity. They can also make in a bulk. No, they can also make in a industrial scale. Okay. So
हाँ
what is
तीन
the
गो
role of
स्पीकर थे
synthetic
तीनों दो हो गया तीसरा
organic
चल रहा
chemist?
है
They will basically
अभी वाइंड
develop
अप हो
a
जाएगा ये
methodology.
खत्म होगा इनको थैंक यू बोलेंगे बॉट ऑफ थैंक देके प्रोग्राम खत्म
It is not an easy task. People think that we can make
हाँ
drugs very easy, but
थैंक दे रहे हैं कुंदन लाल
real synthetic chemists know that transformations of even one new reaction or new functional group is not a very easy. That is a very, very challenging. If you have a new functional group in any molecule, then very well-known reaction
बीच
may
में हाँ
not.
थोड़ा देख लीजिए फिर
So
हो गया
every new molecule
हाँ सर
basically
लास्ट
pose a different kind
नहीं
of
सर
problem. Okay.
नहीं अभी सर
So
पूरा
to
ऑलरेडी
develop the
आपका
methodology
नाम
for
छप गया
functional
सबका
group
फोटो लिए हैं हम
transformation, develop a strategy for the total synthesis of
आपका
methods
कॉलेज
where
पूरे हिंदुस्तान में टॉप हो गया
synthetic
सर
chemists can
ये चीज
also
आप तो
basically
फेमस हो गया
investigate the interactions of synthesized compounds,
हो गया सर
potential
बहुत
drug targets,
टॉपिक से
and
इतनी
most
इतना हो गया ना
important is to develop a method to scale up. Laboratory synthesis of organic molecules and
हाँ
bulk
बैठे
scale
बैठे बैठे
synthesis is totally different thing.
अब 10 मिनट होगा ही
If you want to make 100 milligram, the process developed to
हाँ
synthesize 100 milligram will be totally different
रजिस्ट्रेशन करा
as
लिए
a
हैं
10
मिल जाएगा
kilogram
जो जो उस
scale
लिस्ट
of
में है
compound.
225
And that will be
सबको
totally
मिल जाएगा
different for 100 gram
देंगे
of
सर एक
or 1
सप्ताह में देंगे
tons
अच्छा
of
देंगे सबको देंगे
that compound.
अभी हर
Okay, so we need to develop a tool for technology to basically synthesize those compounds in a bulk scale.
लोग अभी भी जुड़
Okay,
रहा है
so there are lots of scope for synthetic organic chemistry. Now, question is after having drugs in market and consider you just made one compound which is useful and acting as a drug, then you cannot become relaxed because we observe many times that those drugs get resistant within 1 or 2 years. See, you use 15 years to develop a drug, and within 2 or 3 years, virus get mutated. They become more powerful. They started showing against that drugs, resistant against drugs. So then here once again synthetic chemists have to play a significant role. They immediately have to start to investigate, make a more powerful drug to kill the virus or bacteria or parasite. Okay. Here just I took one example of penicillin. Penicillin basically developed in 29, and the structure of the penicillin was this one. If you see this slide, the first structure is basically was basically used during 1929, and this compound was used for treatment of different kinds of bacteria. But within years, bacteria got mutations, and due to that, synthetic chemists had to develop different kinds of molecules which can basically kill that bacteria. So here, these are the derivatives of penicillin or higher generations of penicillin. So this is the first generation, second generation, and third generation, then fourth and fifth generation. So at present, we are using the fifth generation of penicillin. So I want to say by taking this example is that after making or developing one new drug, if you feel that the we basically solve the problem, then it is not good. You have to always keep monitoring different kinds of patients, and you have to keep monitoring the virus or bacteria or different parasites. And accordingly, you have to devise different methods to make more powerful drugs. So here I just want to take one example which basically I developed when I was doing postdoc. basically I make a basically I did basically altering one very well-known antiviral compound that is called SGT. So SGT is basically very well-known retrovirus compound, and if you see, this is basically a nucleoside kind of a compound. So this is basically the oxynucleoside, and at the 3' position, in place of OH, you have an SI group. Okay, so this compound was basically used for treatment of SGT. So, but once again, this compound started showing resistant by virus, and after few years, this compound become useless. So now organic synthetic chemists started to develop more powerful basically drugs so that we can combat HIV. So basically we also started to make more or less similar compound that is called 4-seleno-AGT. If you can see, basically in place of oxygen, we basically replaced selenium. We thought that by having selenium, there will be more proper binding against the enzyme associated with virus, and that may kill more properly. Okay, so these are the steps. Basically, I'm not going to discuss the chemistry behind the synthesis of this compound, but here I put this synthesis in one slide. But I promise here that this seems very easy, but we took 2 years to complete the process of synthesis of this post-seleno-AGT. Okay, we started with readily available deoxy-D-ribose. We developed different kinds of method for cyclizations of selenium, or making this kind of chironoside. Okay, and then we developed different kinds of methods to introduce this kind of base to the chironoside, and then we brought finally this compound. But after making this compound, when we strained this compound against HIV, we found that this compound is totally useless. This compound is not active against HIV virus. Then we tried to find the reason, then we found that the structure of this compound is totally different as compared to the structures of corresponding real AGT. If you see the 5-member ring, the 5-member rings has basically having conformation that is called short conformation, but in AGT, 5-member is generally having the knot conformation. So just by putting one simple element or replacing one element, oxygen to selenium, the basic property of compound is totally changed. Okay, although this compound was not very useful against basically HIV, but that gives idea that for making a good HIV agent, okay, HIV drugs, you need to have a lot kind of conformation of 5-member ring. So this gives a lot of idea, and we basically further improved. And at present, I'm not going to discuss, but this information basically gives you idea that although we can make drugs, but we are not sure that that drugs will work for a longer time. We have to keep improving those drugs so that we can combat all kinds of virus. Okay, so here basically I want to just stop by just giving this statement by one good scientist, that is Neil Kager, that synthetic chemist legacy in addressing past crises is very impressive, yet the effort is never complete. It developed a lot of methods in organic synthesis, but still we have to do a lot of things. So we have to keep going, keep improving the organic chemistry, and if we get any kinds of natural crisis, then we should be always ready to combat those kinds of crises. So once again, here I would like to thank Dr. Ujjwal to giving me opportunity to present this basically webinar, and thank you to all of the audience for your kind listening. Thank you.
Thank you. Thank you, sir. You have presented a very informative lecture, and you have discussed about drug development, clinical trial, repurposing of drugs, biological studies of library of organic compounds, and also many medicinal compounds, medicines like penicillin, cephalexin, amoxicillin, cefalexin, who and these drugs are highly useful now. You have presented very nice lecture, highly informative. We learned. I think all the participants got many ideas about medicinal compounds and medicinal chemistry. Of course, chemist has big challenging role to develop such compounds which is useful for the treatment of any disease. This present time is highly challenging and highly dangerous time for globally. So we can definitely we can find we can get drug after some time. Present time, 2-3 days back we got news from news. Russia is going to launch vaccines. So definitely after some time the remedy of this COVID will come and will get rid of this bad situation. Definitely will get. Thank you so much, sir. Now
I have one question as well, sir.
Yes.
Myself, Dr. Kunal, Department of Zoology, SPR College, and being a geologist, I am very much eager in field of biochemistry. So my question is from all three speakers, Dr. Rafiq Islam sir, Dr. Vishwadipar Tripathi, and Dr. Girish Chandra sir. My question is, in my study, I have found in Patna, especially in Patna, there are a lot of COVID cases, COVID positive cases, but not a single case found in the temple, especially in the Shiva temple. No priest, no individual residing in the temple is infected from the COVID. So my question, I think our field in Bihar, rather North India, have full potential of such plants which are very much capable to prevent the COVID-19. Why, being a chemist and biochemist, we are not working on our plants? In Susut Sangita, one line, I want to put one line. If nature is providing you disease, only nature have cure. So my question is from your all three speakers. We have lot of potential in our indigenous plants. Why not we are working on our plants, our indigenous plants, so that we can find a very raw drug regarding COVID-19?
May I answer?
Thank you very much, sir. I will try to answer a little bit this question. First of all, as much I understand, you are asking for our traditional system of medicine which we were using. Why we are not using our traditional system? So the one reason which I feel behind this, that the fall of our traditional medicine system like Ayurveda is the primary reason is that Ayurveda is the immediate cure. So if some disease like COVID-19 we encountered immediately, so it was new for all the world, and no one was not knowing. So in our traditional Ayurveda, we get a system for prevention for how we can prepare our body. That is for Ayurveda stands for that which increase the age of person. But for immediate cure, Ayurveda is not sufficient. We have to find some different medicine system which is giving the symptomatic relief of immediate cure. The other reason what I feel behind the fall of this Ayurveda or traditional medicine system, the literature which exists in Ayurveda, that Vedic Sanskrit or something, most of the literature are in different are language which are very difficult to understand, or which language which is eliminated from our education system. So most of the person or intellectual group who are involved in the drug research and all these things, they are divide off or keep away from the a big literature or big language what is written there. Other reason what I feel that what you was talking Sushrut Sanhita or Charak Sanhita, what was that? At one time it was at its peak because I am very much related with this field. I was reading a book, Chikitsa Chandrodaya. It was around the 1700 something. That was the book of my grandfather. It was kept in my house. When I opened the book, I opened a chapter of that Unmad Rog. It was written in Hindi. That was a CNS active agent. And when I started to reading the treatment, what it was discussing, it was first it's the place, the height from the sea level, the temperature, and the position of moon, position of sun, longitude, latitude. When everything is fixed, then it is starting its treatment. So you can imagine what was the level of research of that person at that time. But that level of research is not continued when the when the medicine research is continued after. Why I think our medicine system is lacking behind.
Thank you, sir. But and supplementary question. I have worked on a plant named Aegis marmelos. And this plant has 35 organic compounds, which are very close to remdesivir or fludarabine. And the properties are almost similar.
Yes.
Those plants who are in daily touch with Aegis marmelos plants, they are not infected. It means the Ayurveda have some distinct property to prevent this disease. So why not we Indians may lead the world to prevent for the major prevention of this disease? Because prevention is the major cause. If we prevent the disease, I am very fond of Dr. J. Chandra's lecture that one plant may cure cancer. If one plant may cure cancer, why not there are many plants which may cure COVID-19 too? Dr. J. Chandra, sir, please.
Yeah, Dr. Sir. First, I will say that actually natural product we are extracting from nature, we are always using for the treatment of different disease. Although we are not directly using, but most of time we are getting inspiration from the nature. If you go through the literature, then most of medicines present in market are almost 70% are basically inspired from the nature. Okay, your question is basically why not exactly we are using Ayurveda compounds for treatment of corona or other disease. Basically, as we basically mentioned and other speakers also mentioned, this drug development either through Ayurveda or by another way, traditional or synthetic way, is basically a very lengthy process. Okay,
Yes, yes.
different stages you have to maintain. Okay, so for that you need a lot of basically collaborations, a huge amount of fund you need, and more specifically you need a government support. Okay. Here, just want to mention one specific medicine, that is artemisinin. This is basically very wonderful, and I will say one kind of a sanjeevani for the treatment of malaria. At present, for treatment of malaria, we are using artemisinin. Okay, this folk medicine is basically came from the traditional literature of Chinese literature. Okay, they basically went back to just investigate different kinds of medicine mentioned in their literature, and they found that basically this medicine is very useful for the treatment of this malaria. Okay, so same thing we can also do for Indian cancer. Okay, but the problem is that and we need for promoting this kind of research is not basically available, and collaborations are also not very available. Although we have different kinds of CSIR labs to basically promote this kind of research. Okay, you are basically eager to promote Ayurveda medicines. And basically, our present government is also basically promoting, although in a very slow pace. But hope you can just hope that, or we can hope that in future we will be in the position that our few medicines from Ayurveda will come to the market and basically lead to the whole world. Okay.
Thank you, sir. Thank you.
Thank you, sir. Thank you, sir, for your nice lecture. Now, I'm calling Dr. Kundalal sir for vote of thanks. Kundalal sir, please.
Okay. So finally, on behalf of organizing committee and SPR College Barbigha family, I am feeling great pleasure for expressing my gratitude from deep of my heart to our chief.
Kundal sir. I think some problem. हाँ सर कनेक्ट होइए हेलो बस लास्ट टाइम है सर यस I think again technical problem has come. So this time for vote of thanks, I think he is disconnected. So I'd like to give my I would like to give thanks to all my distinguished speakers who has given very important time to us for conducting this program. So in my college, infrastructure is highly poor. So in this situation, we organized, and also I'd like to give thanks to all staff members of this college and faculty members who have supported me. So finally, this program became highly successful. Thank you so much, sir. Thank you so much, sir.
Thank you, all of you. Thank you very much.
Thank you, sir.
Thank you very much, all of you.
Yes, thank you, sir. डिस्कनेक्ट हो गया, कुंदन लाल हो गया डिस्कनेक्ट, नहीं दे सके। हाँ हेलो, हाँ सर हमने थैंक कर बोल दिया, हमने वोट ऑफ थैंक्स दे दिया। हाँ अच्छा कोई बात नहीं सर, हो गया ठीक है। हमने आप प्रोग्राम को कंप्लीट कर, डिस्कनेक्ट हो गया प्रोग्राम फाइनल। हाँ उसको नहीं हमने लिख दिया, वो मैसेज में कर दिया है, उसमें 6 to 7 डेज के बाद फीडबैक फॉर्म मिलेगा। इसमें नहीं सर्टिफिकेट फीडबैक फॉर्म आज रात में देंगे, सर्टिफिकेट 6-7 दिन के बाद देंगे, हमने लिख दिया। ठीक है सर, ठीक है हम थोड़ा स्पीकर से बात कर लेते हैं थोड़ा अभी, ठीक है ठीक। अच्छा डिस्कनेक्ट हो गया सर, लिंक तो बैठा, इसका आपका मोबाइल में डिस्कनेक्ट हो गया उसी टाइप से हुआ। हेलो थैंक यू सर। सर लास्ट में जैसा डिस्कनेक्ट हुआ कि वोट ऑफ थैंक्स इनको बोले थे देने, वो नहीं हमारे कॉलेज में पुलिस सब आ गई थी सर। हाँ भाई लास्ट में हम ही देना वोट ऑफ थैंक्स, अब तो बोलना था क्योंकि पुलिस सब भी, अब जो भी था मेरे समझ में आया वो तो पीछे में डिस्कनेक्ट हो, 2 बार डिस्कनेक्ट हो गया था। अच्छा जी हाँ सर आप लिखे हैं ना सबका, आप हमको लिख के जरूरत दे दीजिए सर। आप दे दीजिए और कुछ बाकी का भी उसमें जो आप अपना भी नाम दे दीजिएगा, ठीक है आप अपना नाम दीजिएगा, हम उसको बात करेंगे। सारे स्पीकर समझे, सब स्पीकर में जो पॉइंट वो दे दीजिएगा, वाइस चांसलर का दीजिएगा। हाँ ठीक है हेलो हाँ सर किए थे सर, बहुत बहुत धन्यवाद सर। लास्ट में थोड़ा सा डिस्कनेक्ट हो गया पटना से ही और हमारे कॉलेज से लास्ट में थोड़ा सा हाँ नेटवर्क का थोड़ा सा सर अच्छा प्रोग्राम, हाँ। और सर बहुत सर हमारे भी यहाँ पर हमारे डीसी सर भी बहुत खुश हुए इससे। और सर हम अब थोड़ा सा न्यूज बनाते हैं, उसको पेपर में दे देते हैं, ठीक ना? हाँ ठीक है सर, ठीक ठीक ठीक, थैंक यू सर। आप बहुत इम्पोर्टेंट टाइम सर अपना निकाल के दिए, जिससे बहुत ही बहुत हम बहुत खुश हैं कि सर जानते हैं सर, जिस जिस जहाँ से हम लोग पढ़ के हैं, जिस कॉलेज में हैं, यहाँ नहीं माने मन नहीं लगता है कि कैसे रहें, लेकिन जॉब है, अपना लाइफ है, इस छोटा सा जगह में अगर कर रहे हैं तो बहुत क्या करें हम। जी 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 जी, अपडेट रहिए, यस सर, यस सर। अभी अभी जानते हैं सर, इस कॉलेज में एक भी प्रोफेसर हमको देखते हैं केवल 30 पद हो रहा है। बस बस बस, हाँ सर, हाँ सर, हम आएंगे सर, नया आएंगे, आपसे मिलेंगे सर। ठीक ठीक है सर, ठीक, थैंक यू सर, थैंक यू। हाँ बोलिए सर। अच्छा सर, न्यूज हिंदी में लिख के दिए हैं। अच्छा सर, आप तो तैयार किए बहुत, आप तो बहुत आप ही कम, ये तो क्या करें हम। लेकिन नहीं नहीं नहीं, बहुत अच्छा, लेकिन एक चीज है, हम रिसर्च हो गए थे आपके साथ, हर सेमिनार हर प्रोग्राम में आप हमको साथ देते हैं। ठीक है सर, ठीक है, इसको तैयार करते हैं, ठीक है, उसके बाद देते हैं। ठीक है। अब अभी उनका वो गिरीश सर का भी फोन आया था, तो वो भी बोले कि बहुत अच्छा आपका एफर्ट देख के हम बहुत खुश हैं। तो बोले कि आप इतना मेहनत किए, हम लोग सरप्राइज हैं कि आपकी छोटी सी कॉलेज में लगा करके इस तरह अपने हाथ पे सिंगल बड़ी होकर कैसे और हम बोले कि नहीं सर, सब आप लोग का जो आप लोग समय दिए, तो और मेरा हिम्मत बढ़ा, और हमारे साथ में और कई हमारे कॉलीग हैं जो मेंशन किए, जो दूसरी यूनिवर्सिटी से वो लोग हेल्प किए। हाँ हाँ, और हम यहाँ भी करते हैं, जो मुंगेर यूनिवर्सिटी का है ना, वहाँ पे हम दे देते हैं, भव्य सर को वहाँ छपवा देंगे, उस उधर लोग। हाँ बीसी सर का फोटो, जो लेक्चर दिया है ना, वो फोटो निकाल लीजिए। अब तो यूट्यूब का रिकॉर्ड हो गया है, वहाँ से जो भी फोटो चाहेंगे फोटो ले लेंगे। और ठीक है सर, ठीक ठीक। बहुत अच्छा आपका लेक्चर, बहुत अच्छा। नहीं सर, कोशिश नहीं, हम रियली बहुत खुश हुए कि छोटे से कॉलेज में, और अगर यहाँ से ऑर्गेनाइज किए, वाइस चांसलर का सपोर्ट मिला, मिला, लेकिन आपका लेक्चर सब बहुत, जो हम चाह रहे थे ना, वो चीज़ आपने नहीं कर रही है, ये चीज़। सर हाँ हाँ, लेकिन आपका जो लेक्चर था ना, वो तो बिल्कुल एकदम ये था, एकदम पूरा कि कोविड तो प्लस वैक्सीन में बिल्कुल एकदम मिलता जुलता है सब कुछ, बहुत वंडरफुल सर। और ये अभी यूनिवर्सिटी से सर बहुत अच्छा। और ये यूनिवर्सिटी से अभी खबर आई है हमको फोन करके कि बहुत अच्छा आपका सक्सेसफुल हुआ सर। थोड़ा सा आप न्यूज़ में उसको बना रहे हैं, न्यूज़ में देख के उसको छप जाएगा। ठीक सर। हाँ सर, श्योर 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 श्योर, आपका तो नाम पहले में है। मिल जाएगा सर, मिल जाएगा, श्योर मिलेगा सर। और सर हम आएंगे, थोड़ा नॉर्मल सिचुएशन में आपसे मिलेंगे हम मोतिहारी में। ठीक सर, ठीक ठीक, प्रणाम सर, प्रणाम। जी जी जी, हाँ प्रणाम। हेलो, हाँ प्रणाम सर। सर कैसे मिला जुला के हो गया सर। हाँ सर, लेकिन हाँ, लेकिन सर आपका ऊपर शुरू में जो आप आए, तो जो कि मैंने कि आप आए और उसके बाद देखा पढ़ेंगे कि सर जो हम चाह रहे थे वो आप, और लेकिन सर प्रोग्राम मिला जुला के अच्छा ही रहा। हाँ, अभी अभी जस्ट अभी खत्म हुआ है 5 मिनट में। थैंक यू सर। सर इसका हम एक आदमी को लगा दिए थे, संजीव झा सहरसा में, वो न्यूज बना रहे थे इसको, हमें लोग के साथ ज्वाइन किए हैं सहरसा एमएटी कॉलेज में। तो वो न्यूज बना रहे थे, वो टाइप करके हमको दे दिए हैं हिंदी में। तो सारा मैटर आपको हम भेज देते हैं, आप उसमें नवल सर का और सब जो हैं, ये वेदर पांडे सर का जहाँ उचित हो जगह दे दीजिएगा एडिट करके। और फिर वहाँ भी रंजीत न्यूज़पेपर में छप जाएगा। और यहाँ पे ठीक वो आप हमको दे दीजिएगा इधर, सेंटर को इधर हम वेंडर सर को दे देंगे, वो दे देंगे यहाँ पर, ये छाप देंगे। हम आपको जस्ट अभी सेंड करें, उसको पढ़ लीजिएगा। फिर जहाँ और कॉलेज के जैसे कि सेक्रेटरी में है वीरेंद्र पांडे सर, इनका नाम देना है, नवल सर का देना है, वो बोल रहे हैं, वो दे रहे हैं। आपसे जल्दी कर दीजिएगा, हम दे रहे हैं आपको। हिंदी मीडियम में है। अच्छा एक बार आप देख लीजिए, मैट्रिक जो अभी है वो कंटेंट ठीक है।